all the panel presentations. But if there's a burning question, I can take it. Otherwise, we'll just go on. Can I now invite Dr. Urvashi Singh, the current Deputy Director General of Central TB Division, Ministry of Health and Family Welfare. She brings with experience in the field of MDR, XDR, epidemiology, diagnostic, biomarkers. She's been a member of the expert committee of CIDESCO, TB elimination program, ICMR, DST, DBT, and WHO. Madam.
So with that vibrant talk, I hope I can do justice. So the topic given to me was a little vast, so I'll try to run through the slides I put together, but then if there are further questions, I'm happy to take them whenever. So this is what was given to me: transforming TB control, vulnerability mapping, and integration of algorithmic solutions with NTEP. But then I was asked also to talk about drug resistance, to talk about, of course, Sir has dealt with the journey of treatment of drug resistance. And, I'll see where I can pitch in my thoughts. Now, this is the summary of our TB patients. So of the set of exposed, we know a good number get infected, but a small number break down. Why we say a small breakdown? Because we actually see disease in a small number. The innermost smallest circle is what the microbiologically confirmed group is. And of course, there are clinical tuberculosis, which may not always be picked up microbiologically. So we have true active TB, which is definitely active TB. We have the larger circle of infected and still larger circle of exposed. Here I share with you the data that was published in the National TB Prevalence Survey, which brought up done by the Ministry of Health, that is National TB Program, along with ICMR. And what this brought out is something we all need to ponder over. This brought out that the country has about 316 TB patients per lakh population. The highest PTB prevalence, the pulmonary TB, highest numbers are in Delhi, lowest in Kerala. But then what it actually brought about is what guided our recent, the campaign, the 100 days campaign we to the program. And it was all scientifically designed. I'll talk about it in a minute. So what the prevalence survey actually brought out was the vulnerable groups. And that is one of the topics they wanted me to speak on. And subsequently, I think there's another talk which talks about vulnerability. I will briefly touch upon why vulnerables are important. In a country so vast, the vulnerable groups, smokers, alcoholics, diabetics, HIV, those who had TB in the past, those who are in contact, and those who are geriatric population, those who are low BMI. 7 to 8 groups are the predominant ones, which our prevalence survey actually brought out. So this is something we cached when we designed our campaign strategy. And the other thing we noticed in the prevalence survey was that 63% of chest symptomatics did not seek healthcare. Also, this brings me to the concept of subclinical TB, which is again an ignored concept for a long time. There have been publications globally. We'd worked on the subclinical TB in the year 2012-13. And, we made some research proposals, never honored. What we showed was, in fact, I'm not dealing with that here. What we showed was in type 1 diabetics, children who were coming to the OPD for diabetics management, more than 10% had culture-proven TB. And since they were asymptomatic, my clinicians refused to treat them. So that has been I would say, the bane of the TB existence, that we want to treat latent TB. We want to treat active TB. We want to forget the gray zone in between. So the gray, what we are missing. So when we talk of missing millions, everybody loves to talk about the missing millions. Blame it on the program, you're not finding it. How to find it? That is why the research where Dr. Gokhale sir is leading the DBT, where a lot of biomarker studies are ongoing. I have been working on proteomics. We have some of our own molecules in my professor's role at AIMS. There are friends here who've been working in the area of proteomics, others working in the area of vaccine, exosomes, others working in a lot of basic research ideas, which, let me promise you, we will take forward the moment you're ready with them. So this was the National TB Prevalence Survey, and this was the National Drug Resistance Survey, which actually told us this is now old, and we are soon coming out with the latest data. Algorithmic approach. So, sir, we have algorithms in place, and let me tell you, all these are very scientifically. I'm going to come to some basic, So this is the, this is taking forward what sir addressed earlier. The success of the regimens that we've had so far, the shorter regimen to the tune of 72 to 78%, the longer oral regimen less. But what we have today is the BPAL-M regimen. The BPAL-M regimen, which came up, globally. There was a lot of recommendations. Several countries rolled knee-jerk response. We in India prefer to have in-country data. So we had research done in the country by NIRT Chennai, which is also our national reference, one of the national reference laboratories. And they, in fact, modified the dosage. So globally, we had several trials. But let me tell you what the country designed. This was the BPAL-M regimen, which we rolled out after I took over as DDG in August. The regimen that was recommended by WHO is also different. What we rolled out in our country is based on TB practical study and NTB study, where recurrence-free cure was brought about when we added moxifloxacin despite fluoroquinolone resistance. So this is what we are doing now. We rolled it across the country. 18 states have actively enrolled patients. And the newer drugs, Benaquilin, Pretomanid, Linezolid, Moxi being always, they were there earlier with us. Benaquilin resistance, let me tell you, is not very high. So we are quite comfortable, not more than 1.5 across the country. Few pockets which possibly have certain genotypes which are known to be resistant. For example, Mumbai has a little more BDQ resistance, but Pretomanid, since we're introducing now, we have equipped all our labs across the country to do phenotypic DST for Bedaquiline and Pretomanid. We are also doing targeted GS for patients who are being enrolled on BPAL-M. Now, coming briefly to some basic research, basic questions. Now, this is not a fresh slide, this is an old slide, but these are the factors which affect the generation of MDR-TB. And the mutations, all of us are aware. Let me move forward, and there'll be somebody addressing this. I want to talk about the epistatic interactions. Sir talked about, he laid the ground for me. So epistatic interactions, all of you may also be aware, maybe, mutations at a far off site, which may or may not affect the fitness of the organism. Those which affect the fitness of the organism do not let the organism survive. Those which have zero fitness costs, those those organisms would survive, and those mutations will be responsible. So here, if you could appreciate the CATG mutation could have other epistatic mutation at the site of AHPC, or maybe other CATG sites other than the 315, which is responsible for high-level resistance to INH. RPOB 531, there are mutations in the RPOC, RPO-A sites, which can be epistatic. Similarly, Amica cineraris 1401 is the one that's considered causal for resistance. Similarly, for Gyre. So moving, I would need to just quickly move through. So this is, again, not new papers, but they talk about synergistically acting drugs, which we think could be synergistically acting, but could be enhancing epistatic mutations, could be enhancing resistance to another drug. Here, there's an example of combining fluoroquinolones with rifampicin, which in the clinic, several people like to do. These would enhance epistatic mutations to one another. So epistasis is basically compensatory mutations which depend on the strain genetic background, and this could affect transmission, level of resistance, amplification of resistance, and clinical outcome. Now, this is just a glimpse to show that high fitness mutations can accumulate in places like congregate settings. This is a Georgia prison, the study from, which says that, high fitness mutations could get transmitted. And it's the effect of drug pressure, which we traditionally know, drug pressure, and how this can enhance more newer mutations. This is, reiterating what I said earlier. Prisons can be drivers of, enhancing or encouraging new mutations, which are high fitness, and of course transmission because it's a congregate setting. Now, this is development of this is bedaquiline resistance. Now, one concept that has not really been delved into is heteroresistance. So at the outset, polyclonal TB, I'm not sure how many of you have actually read polyclonal TB. We published polyclonal TB in DSTB in 5 years back. The polyclonal TB is basically combination of different genotypes. In MDR-TB, that is a very well-known concept published, world over. But what we showed in 2020, we published that even DSTB could be made of mixture of different genotypes, and heteroresistance, hence, on treatment, heteroresistance could easily build up. Now, I'll bring you to, this is, during treatment, this is a patient, this from Nepal, he reached Switzerland, possibly could pay for it. And you can see while on treatment, he accumulated several new mutations and resistance came up. Now, the key challenges for the program, I'm mixing up different concepts because that's how they wanted me to touch on. Dr. Gambhir asked me, touch on this, touch on that. So I'm trying to do my best. And so the challenge we faced is to reach the unreached. I talked about congregate settings, but also slums, tribal, and vulnerable. The drug-resistant TB, of course, it's very important to diagnose early. Initially, at the outset, we are offering universal DST. I'll come to that briefly. Comorbidities like HIV, diabetes, I alluded to this when I said prevalence survey, brought these out. Undernutrition, low BMI, overcrowding, which will enhance transmission. And of course, like I said, carry forward highly fit strains. So what we did in the campaign, was we picked up the 3 or 4 strategies quickly. I'll share with you. We identified, since vulnerables, I did not get a picture of our NICSH portal, how much vulnerables contribute, but offhand, the maximum contribution is by the low BMI. 46% of our TB patients under treatment by the program have BMI less than 18.5. So that is something we really need to work on. So what the program did was, from 1st November, the direct benefit transfer scheme was doubled from 500 to 1,000 rupees per month throughout the treatment course. We recommended energy-dense nutrition support, a study we conducted at AIIMS at the Balabgarh, where we showed energy-dense nutrition support for initial 2 months. 2 patients who had lower than 18.5 BMI made a lot of difference. Reduced mortality, the treatment response was better. And Nikshay Mitra is a scheme our Honorable PM launched in 2018, which has been expanded based on the ration study model to the families. So now the CSR support for Nikshay Mitra baskets is being given to families, along with the patient, taking care of our undernourished patients. But what we possibly could also do is actually look at population level undernutrition reduction. Vulnerables, so this was a nutrition initiative. Amongst the vulnerables, what we did was we started doing active case finding, screening by X-ray, confirmed by NAT. So we have a wonderful network of fixed X-rays. We also enabled our program staff, the peripheral level workers, with handheld X-rays. Screening was offered to all the vulnerables that I mentioned earlier, followed by confirmation by NAT, which again, we have our strength. We have more than 8,540 systems in the country, NAT systems, which will pick up drug-resistant TB, at least MDR-TB, RIF resistance. But we are also now soon getting systems, POC, made in India, indigenous technology, plus rifampicin, INH, and fluoroquinolone. So our drug-sensitive patients will be taken care of by these systems. Of course, for now, we're using line probe assays where AI reading is also being used now, not really implemented countrywide. So we took care of this, and we took care of our vulnerable and overcrowded, our congregate and overcrowded settings. We went to slums, we went to prisons, we went to old age homes, correctional homes, and so on and so forth. And we still plan to strengthen this further. Lack of awareness and poor health seeking behavior remain some challenges which are beyond the program and where we are seeking help from others. Now, this is a study since I was asked to touch upon the latent TB infection. This is an Indian meta-analysis. The gist of this is that reactivation disease has been overestimated, and the TST positivity, or for now we have the eGRAs from, they've been around for some time. And what we have in the country is CyTB, again, an indigenous technique, which the technique it adopts from TST and the antigens it adopts from eGRA. So this would actually pick up CMI. Since we have basic researchers here, I would actually urge you to tell me whether just demonstrating a CMI to TB is indicative whether the infection is present today, or was it present yesterday and it's no longer there. That is, I think, a misnomer. And hence the LTBI is overestimated. Biomarkers is possibly the omics technologies, the omics research needs to guide us where there is infection, where there is disease, where there is subclinical disease. So when I talked about the campaign strategy, must bring in the subclinical disease. We went also to asymptomatic vulnerable. So as to pick up disease early, we pick up subclinical TB. The definition of subclinical TB is asymptomatic, possibly culture positive or possibly radiologically positive. Now, briefly to touch upon the AI, which was my original topic, where all we are using AI. Of course, we're using AI for reading X-rays. The handheld X-rays in the periphery do not always need a medical officer, the healthcare workers. With the help of AI solutions, we have IPR technology recommended by ICMR. We have Cure AI with us. Several states are using, one of them. Which can actually tell us, so what we want to do, worker should not initiate treatment on the AI report of TB, because that will pick up past lesions. And that is when we move from the National TB Control Program to revised National TB Control Program in the year 1992. We did not want to base our initiation of treatment based on X-rays. We wanted it to be done based on microbiologically active disease so that the treatment will be given properly. We need the AI to tell us normal or abnormal, and the abnormals are then further tested by the globally recommended NAT technology, and then we initiate treatment. This is AI technology for predicting adverse TB outcomes, and I'm rushing through this here. We have AI for picking cough. Now, this is something which was the topic also given, where vulnerability mapping, in addition to vulnerable groups, what we also plan to do is look at vulnerable geographies. So this AI technology will give us villages and hotspot pockets in the villages. So this is, the micro planning, how it can actually help us reach the areas which may have high transmission. And so I wish to thank you with, the messages should be all around. What we know, we should not conceal. Like I was frank enough to say, whether, looking for CMI. And of course, the program welcomes all suggestions, all thoughts, all ideas, and frankly, research in the country. So kudos to all the basic research scientists who are here, and thank you for your, hard work. And we'd love to take up any new molecules, any biomarkers, and of course the sequence adopted targeted NGS for a BPAL-M, but what Sir is revealing today is the 10,000 genomes, where again our NRL, Dr. Siva, is here. He's supporting us in extending targeted NGS, which will give us the drug resistance mutations known to be, that can tell us which drug to use, which drug not to use. And, during the treatment, if there is, accumulation of resistance, like I said earlier. Thank you so much. And if questions, I can take it.
Thank you, Dr. Urvashi. That was indeed a panoramic view of all how we can move forward. Thank you very much. Can I now request Dr. Sheetal, who has been for many years working on host pathogen interactions and what it means. I think the challenge always is to look at both sides of the coin. The easier side to look at is the microbe, easier relatively. The challenge is in the complexity of the host and try to needle pick what's more specific for the microbe and what's happening. So with these few words, Dr. Sheetal.
Thank you, Dr. Balganesh. And I also want to thank Dr. Urvashi for pointing out several of the things that are going to be revisited in my talk. Thank you for laying the ground for all that I'm going to talk about. So do I have yeah, okay. So I'm going to talk about comorbidities, which are the gray area or the real burden of tuberculosis. And before I do that, I want to really point out differences between what we mean by risk factors versus comorbidities. So we've heard diabetes, HIV, malnutrition, smoking, poor air quality, alcohol use, depression, stress. All of these can be risk factors for acquiring tuberculosis. But what happens in many cases is that tuberculosis presents with diseases such as diabetes, malnutrition, a condition, not a disease necessarily, COPD or poor lung function, and even depression. And these can be driven by tuberculosis or tuberculosis treatment itself. So let's take a look first at diabetes, which is also known as a twin epidemic. So diabetics, we know, have 3 times higher risk of acquiring or developing TB. If you look at the graph at the top, that basically shows a projection of the increase that we expect in the number of diabetes cases over the years until 2045. And if you look at the table below, I'm sorry, it's a little difficult to see on these screens, but the second largest burden is going to come from India. And that's almost a 40 to 50% increase in diabetes burden that we're going to see in the years to come. Why this is dangerous is because there is worsened glycemic control in individuals who have tuberculosis as well. On the other hand, patients who have TB and diabetes at the same time present with increased disease severity of tuberculosis. So you have more weight loss in these individuals. You have more severe lung involvement, cavitary lesions, and difficulty in treating these individuals. We see more treatment failure. So there is that leads to 2-fold higher increase in mortality. There is greater drug resistance, leading to 2-fold higher incidence of drug resistance. And there are diagnostic failures, and that primarily when things like IGRA are used, because there is a generalized weakening of the T-cell immunity in diabetes. So it's important to look at what is the underlying reason for this comorbidity, right? And before I get to that, I want to actually talk about an allied interesting thing that came up in the TB field, not looking at TB and diabetes, but looking at autophagy and tuberculosis. So that's where basic research and the strength and the positives of doing sometimes in unallied questions can bring a lot of benefit. So for example, there was a group of researchers looking for FDA-approved drugs which would lead to control of tuberculosis. And what they ended up finding was this drug called metformin, which is used for glucose lowering in type 2 diabetes. Why this was very exciting is because it was initially shown to promote bacterial clearance. It improved tissue pathology, and retrospective analysis of diabetic and TB patients showed that individuals who were on metformin actually responded better to. So in India, there was a trial conducted which showed that metformin, introduction of metformin, and mind you, this was done in non-diabetic individuals, it tended to lower the inflammatory markers and decrease in cavitary lesions. However, there was no benefit found to bacterial burden. So while there is some answers that we've got from looking at a drug that is used against diabetes for the treatment of tuberculosis itself, there are still many unanswered questions. And more recent research has showed that there is actually difference in the lipidemia or actually the mice that have TB and diabetes show extreme dyslipidemia. And we probably need to look at things like dysfunctional macrophages and T cells in these granulomas. We need to target dyslipidemia in individuals with TB and diabetes. So these are some of the areas that might show answers to how to tackle this comorbidity. I'm going to now move to poor lung function despite TB treatment. So what's been shown through several studies, and this is actually a meta-analysis showing that the pooled prevalence of COPD in patients with prior pulmonary TB, even after so-called cure for TB, is 21%. So an individual has a lifelong of having poor lung function in a large proportion of TB cured individuals. And the odds ratio for this is as high as 2.53. So again, why is this so? This is thought to be because normally in inflammation is a self-resolving phenomena in the body, right? So you have an increase in inflammatory molecules and inflammatory cells that come to the site of infection, and then there's a resolution phase. But somehow in tuberculosis, this resolution phase is lost in many individuals. And as a result, you have this prolonged inflammation, which can contribute to things like bronchiectasis. It can lead to occlusion of the alveoli, and that might be contributing, that inflammation might be contributing to the long-term effects of tuberculosis that we see on COPD. Ma'am also talked about the rations trial. So I want to talk about malnutrition as not just a risk factor, but possibly also a comorbidity in tuberculosis, which is that it becomes difficult to treat individuals who are malnourished even with nutritional supplement. So this rations trial was an excellent example that when you give nutritional support, not just to the index case, so the blue, the index case in the control arm, but if you also start treating the family members with nutritional support, you can actually decrease the TB incidence in the community by 50%. So this is as good as any vaccine so far has come, right? Nutrition is as good as any vaccine that has been shown of late to provide protection. So this is very, very promising data. But unfortunately, what this study has also highlighted is that this nutritional support is not sufficient for bringing about the that is necessary to see positive results in individuals. So we need to work more towards possibly getting a more specific micronutrient or inflammation-reducing dietary supplements that could help these individuals in not only preventing the occurrence of TB, but also lowering the morbidity that is caused by inflammation in tuberculosis. Finally, I'm going to talk about the syndemic of TB and depression, because this seems to be an area that has been totally ignored, while it is one of the most glaring problems in the world out there. 48.9% of TB infected individuals show depression. This leads to loss to follow-up by about 3.4 to 3.9%. And the overall incidence rate of depression is much higher in individuals who have TB compared to the general population. So what this ends up doing to the patient is the patient is becoming non-adherent, leading to drug resistance. What could be the mechanisms for this? I'll talk about that. But before that, I would like to point out that this has been recognized by WHO as an invisible burden for people with tuberculosis. And what could be the mechanisms? It's been proposed through several studies that TB infection, the inflammation that is happening in TB infection, might itself be driving changes in neurotransmitter levels as well as their functionality, leading to depression. Depression can drive TB infection because depression is known to lower the Th1 immunity, which is so important for control of infection. TB treatment itself can actually have interactions with drugs that are given for depression. Tonomide, for example, itself leads to depression, but INH can actually lead to reduced serotonin uptake, which can lead to increased depression. All of these factors leading to non-adherence and drug resistance. So again, I'm going to here talk about an example that didn't come about by looking for a cure for TB-associated depression, but came into existence because someone was looking for a way for lowering inflammation in TB infection. So this work was done at IGIB. So here we found, so this is work from Dr. Vivek Rao's lab. I was also part of this, where sertraline, which is an FDA-approved antidepressant, was actually found to lower the inflammatory response that is generated in tuberculosis infection. It not only targeted the inflammatory response, but it actually showed synergy with frontline anti-TB drugs, and it could target drug-sensitive and drug-tolerant bacteria as well. So we've gone on to show that this works very well in reducing pathology, not only in preclinical mouse models, but also preclinical models of guinea pig infection. And it also shows that there is no detrimental interaction with the frontline TB drugs. So maybe this could be a solution, but of course, more work needs to be done on looking at whether this is going to solve the syndemic of tuberculosis and depression. So through my talk, I really wanted to point out that we basic researchers tend to look for answers based on what interests us in the TB field, which is for me, lipid metabolism, granuloma, inflammation. People could be looking at autophagy, lysosome biology, and all of these, when we are actually finding ways to make the patient life better, where they can thrive, they can be more vital, and improve treatment adherence, improve the outcomes of these individuals for a sustained period of time, and hopefully reduce the burden of TB sequelae, which is also a huge thing. So with that, I would like to thank you all for listening, and thanks DBT for the opportunity to speak here.
Thank you, Shita. It's one of those areas where we know so little to be starting to open up new vistas. Our next speaker is Dr. Ashwini Kumar, Senior Principal Scientist, DBT and DBT Wellcome Trust India Senior Fellow. He has a number of prestigious publications and also has received the Shanti Swarup Bhatnagar Award. All yours, Ashwini.
Thank you, sir. So today I'm going to talk about the mechanisms that are there in drug resistance in tuberculosis. And the first problem that comes to anybody's mind with tuberculosis is that the treatment is very long, 6 months of treatment, and it's called short course therapy, which is kind of paradoxical, kind of different. The reason for this is this treatment is even shorter. Earlier, the treatment was ranging anywhere from 9 months to a year, depending on the physician who was giving the treatment. And then WHO came up with a guideline of approving the DOTS therapy, and that's where this treatment came in. And this treatment has multiple aims. The first was that unified treatment for all the people. The second bigger aim was the compliance is taken care of so that AMR does not arise, the drug resistance does not arise. And around the same time, some papers, I don't know whether you can see these correctly or not, but 1994, there were some papers published suggesting that with the advent of DOTS, we will be taking care of the rising AMR, and it significantly reduces the drug resistance in case of TB. However, this is the spectrum of drug resistance that we still have. Around 4% of total TB cases, they are showing multi-drug resistance, mostly because rifampicin resistance detect. Rifampicin resistant TB is detected, and if it is also resistant to isoniazid, we call it multi-drug resistant. Then there is a pre-XDR, which is a new term which was coined by WHO in 2021, where the MDR bugs, they have resistance to moxifloxacin or levofloxacin. And then we have XDR drugs, which are basically resistant to rifampicin, isoniazid, moxifloxacin, levofloxacin, and one of the linezolid or bedaquiline. So there is a term which is frequently used in TB field, which is totally drug resistant TB. However, WHO has not approved it because in most of the cases, not all the DST is not done for all the drugs, but there are patients which are basically left with no treatment. And their treatment is the older things, pneumothorax, puncturing their lungs and so on, removing the part of the lung and so on. And it happens. There are few cases, documented cases of this as well. The problem of MDR is this, that if I try to put one pill over another, I will get a building which is 3 times taller than Qutub Minar. And it's a bigger killer. With the drug sensitive TB world over, from 83% to 91% success rate is there. before 2016, the success rate for MDR-TB was only 52%. When I say success rate, at that time, the treatment was 2 years. When I say success rate, that meant that these people have been able to live for 2 years and they have completed the treatment. For XDR, it was even worse, 28%. But then WHO approved bedaquiline, pletominid, and for treatment. And now we have also BPAL-AM, which is having moxifloxacin. So treatment of MDR-TB with these 3 drugs is reduced 9 months. And this is what it has done. From 50% success rate to 68% now. And it is the graph, if you see, with the time is increasing, suggesting that we perhaps can have achieve higher success rate for the drug-resistant TB. So what is the status world over? Where do we stand? So we stand here. We are among top 30 countries which has highest burden of MDR-TB, and it varies. In cities you have more of MDR, in other places you don't have that much of MDR, and so on. So we have to take care of this problem of MDR. And what are the mechanisms? These are the mechanisms. These are mechanisms that are there for almost AMR in everywhere, in other bugs, in other pathogens. The first one is that you modify the target. The classical example of this is rifampicin. You modify the target, which is the RNA polymerase beta subunit. For isoniazid, the bacteria tends to modify the targets. For bedaquiline, ATPE can be modified, but this mutation is not selected as much because there is too much of fitness cost associated with this. And we have seen very little, anyways, bedaquiline resistant strains are sequenced in very few numbers. So we have seen very little of ATPE mutation. For ethambutol, EMB, ABC, CAB can be mutated. And for moxifloxacin and levofloxacin and other fluoroquinolones, the bacteria modifies the GAIR AB. The other is for MTB. MTB is special because a number of drugs, actually prodrugs, and they have to be converted into the drug inside the bug, and then they act. So the bug can modify the activation of the drug. It does not allow the conversion of prodrug into drug. And for that, the examples are that MTB have for isoniazid resistance, MTB has a lot of mutations in SCAD-G for A. And the other example of modification will be ethambutol, ethionamide, where the prodrug is to be converted into the drug. The other strategy is that you basically pump the drug out. So there are a lot of efflux pumps which can be used by bacterial cells to pump out the drug. And yeah, okay. All right, I can go ahead with that's fine. Yeah, so these are the normal drug resistance mechanisms that are there, and we know that these mechanisms exist for other pathogens also. But what is happening is, most of the time when you talk to a doctor and you talk about drug resistance, they say the resistance is because of poor prescription of the drug or because of poor compliance of taking the drug. But with DOTS, we have taken care of that. We see that patients are taking the drugs. We have ways to monitor it. Despite that, there is a lot of drug resistance that is happening. So what are the other factors contributing to it? Recently, there is a lot of literature emerging. There are 2 things that can actually lead to drug resistance. One is differences in the pharmacokinetics of the drug in different individuals. One of the classical example of this is when you have a patient who is metabolizing a drug more and is putting it in the secretion, and so the availability of drug is very little. And towards that, Dr. Rajesh Gokhale's group has published a paper recently, which was published in eLife. That paper actually shows that MTB, when it infects mice, and you can see this in humans as well, the bug moved to the liver. And in the liver, it was known that it can infect Kupffer cells, but now they show that it also infect hepatocytes, which are the real factory of the liver, where the lot of metabolism happen. So by doing that, what they do is they upregulate PPAR gamma, and they themselves become drug tolerant. I will talk a bit more about drug tolerant bacteria also. Besides that, what they also do, the MTB infection also does is that it upregulates the drug metabolizing genes, the NAT, the ICEs. So with that, MTB ensures that the drug that a patient is taking that is metabolized faster is secreted out, and because of that, there can be emergence of drug resistance. So this is a new hypothesis which is there. Besides that, I talked about a bit about drug tolerance. So drug tolerance is a phenomena when you treat a drug-sensitive bacteria with drugs, few bacteria around 1% to 0.1%, they don't die. And now there are experiment. If you expose this, if you repeat this experiment that you treat with the drug and then take away the drug, again treat with the drug, then take away the drug. So what happens is many of these drug tolerant bacteria, they become precursors of drug resistant bacteria. So understanding the mechanisms of drug tolerance is becoming very important. In that, some work my lab has done where we show that in the biofilm there is a lot of drug tolerant bacteria, and then there is a paper from Keith Derbyshire in Science which shows that there is a lot of genetic changes in the biofilm. So so for example, if one become resistant, it quickly transfer that same piece of DNA to the other bugs, and so that the whole population can become drug resistant. And we also did some more work, and we showed that TB, which is a chronic disease, actually is a biofilm infection, and we can detect the TB bug in the form of biofilms in the human lungs, in mice lungs, in macaque lungs, and so on. So these are few mechanisms through which drug resistance can be acquired. DOTS was the first example where the therapy was observed and ensured that the patients are taking and are compliant. Despite that, we see emergence of drug resistance, suggesting there are newer mechanisms that we need to discover and we need to figure out. And Dr. Urvashi Singh has data on betaculin, that betaculin resistance is also emerging. How do you really look into betaculin resistance? So my lab has done some work. I'll just go to that quickly. Yeah. So betaculin is the new drug that has given us a lot of hope, and it binds with the ATP synthase, and it binds to the ATP synthase C subunit, which is in the proton pump from where the proton enters. Right here is the ATP-C subunit, and there are 9 of these. These form a ring, and that ring proton moves. This moves, and that movement lead to conformational change in this subunit, which leads to ATP synthesis. And what betaculin does is betaculin binds right here. There is a Nature paper in 2012. So betaculin binds to ATP-C subunit, and some of higher concentration of betaculin can. So this basically inhibits the movement of this motor and ATP synthesis. When you use higher concentration of betaculin, then what it does is, besides binding to this also binds to this alpha subunit and the E subunit, and then basically it disconnects the ATP synthesis with the proton movement, and that leads to the killing of bacteria. So what we have done is we have performed a transposon mutagenesis, and we've sequenced. And besides the non-target, which is MMPL5, which is predominant in the GWAS studies, and which has been shown, what we found was interesting. There is a gene called as CMA2, which is involved in cycle propagation of mycolic acid, that actually controls the fluidity of mycobacterial membrane, and that is responsible for drug resistance. There are other pathways in this which are responsible for sensitivity, and you can make molecules which can enhance the killing activity by betaculin. And this is what I want to emphasize, and this will be my second last slide. So with cyclopropionate mycolic acid, the fluidity of the membrane is controlled. And when in something like H37RV or the lab MTB, you have high rigidity, because of which drug enters into the cell but does not go out. However, if you mutate this gene, drug enters and come out, and then bacteria display resistance. These results are important because when you compare the sequence of CMA gene across species, betaculin is used for NTMs also. Across species, what was beautiful is that this sequence, the active site is conserved in some, does not have that conservation. So they probably have an enzyme which is not active. Example of that is M. smegmatis, Xenopi, abscessus, and Shimodi. And in all of these, there is drug resistance towards the betaculin. And if we put the MTB-RV's gene back in these organisms, they become sensitive to betaculin. So we believe one has to understand more of these mechanisms of drug resistance, and then we can perhaps figure out ways to even kill the MTB which is resistant. So yeah, that's all for today. Thank you.
Thank you, Ashwini. Interesting. I published this on the cyclopropionate.
Yes, no, it's not
Right.
published.
Brand new. Thank you very much. Sorry.
Yeah, so just give me.
What do you want me to do? Keep quiet? Okay, all right.
If there are questions, maybe.
Yeah, that I'll do. So in the panel A, we've had discussions regarding the complex landscape of tuberculosis, the urgent crisis of resistance and the mechanisms which are conferring resistance in Mycobacterium tuberculosis. We've looked at the efforts of National Tuberculosis Elimination Program towards eliminating TB. Of the chair, I would like to invite questions, if any, and then we'll move on to the panelist B. Yes, Dr. Shankaran.
Yeah, please.
My question is to Dr. Sital. You have mentioned that there are comorbidity of various diseases along with the TB. Have you measured any decoupling effect? Because what I know is that there is a one method, difference in difference, which measures the decoupling effect when several parameters affect a particular variable. But to my knowledge, in public health analysis, this difference method does not work. So that is the first question. Second question is that you have told that nutrition status also has some relevance for elimination controlling of TB. You gave an example of member with the family when the nutritional support has been given, and only the individual in the family where the nutrition support is given, the reduction is from 11% to 5% as per your slide. The whole system of ICE mechanism is that whenever you changes the nutritional direction, it has effects on several diseases. So have you make any study? What are the interaction effect with respect to nutrition study?
Ask the first question to Dr. Urvashi. Sorry, I'm going to pass the first question to Dr. Urvashi for an answer. I will, however, answer the second question. So in that trial, they were trying to see whether there are benefits in reducing the overall burden incidence TB endemic situation by providing nutritional supplementation to individuals who are at risk. So these are not the index case, but family members of the index case. And in that case, they have seen a reduction in the incidence of TB.
So the rations trial was designed around supporting the patient and the household contacts. So this was done in the interiors of Jharkhand, where the BMI was very low. They were able to show reduction in incidence in the contacts. It was much lower than they didn't have a control in this particular study because they could not deny food. But so statistically what they showed was there was a clear-cut reduction in incidence to the tune of about 43%. Also, they calculated the reduction in mortality in the TB index patient. So that was also shown to be significant. And we actually, used this was not the only study in India, but incidentally, this has been published well and, discussed widely. There are more studies from Pondicherry under publication, part of the report. Dr. Sonali is here. Dr. Sonali is the lead. Studies from Chhattisgarh, studies from Odisha, NIRT has done it. Then studies in Delhi AIMS. I talked about the EDNS study where nutritional support. So the demonstration of 5 kg weight gain in 2 months' time was shown in ration study. Similarly, in EDNS study, a clear-cut 5 kg gain in weight was shown in 2 months' time. So the two studies had the similar achievement. And the reduction in incidence shown by, so basically both the studies where nutrition intervention was done, it was only to supplement, not really to. So in the ration study, they used local food, sattu and things like that, which is preferred in the local culture. And in EDNS, we only selected low BMI population less than 18.5. We were given energy-dense, protein-rich, peanut-based food. We've published the feasibility part. The second part is under communication to clinical infectious diseases. Did I miss a part of your question? The first question you asked, she's bounced to me. Could you say it again, please?
See, what is happening is that there was no diabetes. The incidence was something. Now diabetes has come. It has coupled with this TB, and we are telling that diabetes and the incident.
So, sir,
Now
let me
after
address here. Let me address it like this. I would not say diabetes has come now. It is just what the eyes cannot see, we do not know of. So we were not really aware of diabetes, which was so widespread earlier. Now we have the tools and point of care tools. We know at the periphery how much diabetes is there. So what has been observed in epidemiological studies, if we see? So we're not talking about diabetes alone. I'm talking about smokers. So how do we incriminate? How do we explain scientifically why smokers are predisposed to TB? It's probably the local immune response or the chemical injury that this smoking could be bringing about, lowering the immune response. Smokers, alcoholics, PLHIV, we are aware the PLHIV immune response and CD4 counts and so on and so forth. Diabetics, again, similarly. Also, the risk groups are, those who have had TB in the past. Then the geriatric age group. So I would sum it all up as immune response. And not only diabetes, it's the immune response. So whatever is actually hitting the immune response, it could actually be several places. For example, why is the mining community or the construction workers more predisposed? Because of their soil exposure or the exposure to molecules that can induce an in the respiratory tract, induce an inflammatory response in the respiratory tract. And of course, the congregate settings is a high burden because transmission goes on. So these are the factors why we could say why the vulnerable population is actually vulnerable. Also the fact if the LTBI was there, if the LTBI was there and the immune response was weaker, the progression to active TB would be much faster and much more efficient. So what we've also seen, like in these two studies, EDNS and rations, and the other studies on nutrition in India, giving nutrition, so lead to undernutrition because of anorexia. So it's a syndemic really, TB and undernutrition. When we supplement TB patients with nutrition, their response to treatment is better. Their pharmacokinetics becomes better. We talked about, several experts talked about PKPD. We have published in pulmonary TB we used to call it category 2 earlier. We call it retreatment now. We published in abdominal TB over the past few years. In patients, we found lower drug levels in the serum. Now we've communicated something where we look for drug levels in CSF and TB meningitis. Not all the drugs were actually reaching the CSF in the right levels. Sir alluded to the same drugs for EPTB. So we need to look at the basic science to define our regimen. And that is what my epistatic question was also to actually highlight. Synergisms have to be, so I always joke with my undergraduates. It's not like it's a sweet shop, you pick up 1, 2, 3, 4. No, you have to know the science behind it. Only then can you answer. You want to deal with PKPD issues? Nutrition is one of the answers. If the undernourished may not be absorbing all the drugs, they may be having GI lesions we don't know of. They can be, malabsorption because of the disease. Where we talk pulmonary is not secluded pulmonary. Similarly, EPTB is not secluded EPTB. 50% of EPTB patients have culture positive pulmonary TB. And they have radiologically positive pulmonary TB only when you're looking for it, sir. So it's a complicated scenario, but the baseline, the basics are, we need to enhance the immune response. We need to keep the immune response optimal. Otherwise, why would the geriatric population get it? They hardly go out of their homes. They're not contacting a TB patient, possibly, reactivation because of the lower immune response. But again, I am one of those proponents who believes in reinfection. That is why I brought this slide where I said TB is more likely to be reinfection. And where the immune response is weaker, it is not able to deal with the reinfection. So reactivation is a concept on its way out, and we'll soon prove that. Thank you. I hope I've addressed
Thank you. Other questions?
Yeah.
Yes, we have.
This is Mukesh from NIPGR. My question is directed to Dr. Ashwini Kumar. So you discuss about this biofilm where like one bacteria is transferring this drug resistance to others which are present in biofilm. So similar study also did by Samay Pandey from ISC Bangalore. So he also shows through nanotube this drug resistance could be transferred to other species of bacteria. So like it's a complex situation in case of MTB, like some new drug resistance can come up from another bacteria. So what could be a good strategy to tackle this situation?
Dr. Ashwin, can I just comment? So traditionally, globally, all the basic research done doesn't go in favor of horizontal gene transfer in the case of MTB. That is something that is not yet proven. So biofilms, I would be of the thought it's the replication or the new daughter bacteria that are coming up. Horizontal gene transfer, unless Dr. Ashwini is going to prove, possibly it'll be a Nobel laureate question.
Maybe nanotube, like it's a nanotube is a physical thing. So I'm not sure.
Let
Yeah.
Dr. Ashwini reply. Yeah.
Yeah, so there is a paper in Science by Keith Derbyshire with so what he has done is he had kept two strains of Mycobacterium tuberculosis. It is not TB, and one has a drug resistant marker, and other did not have the drug resistant marker, and you can mark these two strains, and he mixed these two and made a biofilm. The strain which did not had the drug resistance now acquired after interaction the drug resistance. So yeah, I know the MTB genome is very stable and there are only few instances of horizontal gene transfer that has been documented. But in biofilm, the exchange of genetic material increases. We still don't know the mechanisms for Mycobacterium because Mycobacteria has a thick cell wall, but it does happen.
So sir, how you can tackle
Can
this
you
situation?
take it offline, please? Because there
Sure.
are others who are asking questions. Yeah, please.
My name is Pawan. I am pre-connected. My sister and TB is diagnosed. Then AKT for kidneys given, right, sir? AKT for generally, like according to our given. So in case of kids, it's very difficult to give because these are four big four drugs. These scientists are planning to design a drug which is easy to give to kids.
Question is planning to design a drug.
Or easy to give to kids. Well, I think you
want to
want
answer.
That question is for Dr. Gokhale.
Anyone.
Okay, so
So I agree with you.
there has been very little work done on that pediatric formulations for TB, but Dr. Soumya Swaminathan actually initiated a lot of work on that aspect. So yes, it's happening. Answering your question separately, has enough pediatric work been done? No, there is actually a big lacking on that. You want to add on that?
Ritomanid is not recommended for children. So we are struggling to get a shorter regimen for MDR-TB in children. There are different combination regimens being, experimented, if I may use the word. Trials are going on for different combination regimens. But yes, the newer drug, Ritomanid, is not yet recommended for children. Bedaquiline is. But again, in very small children in India, we're not able to use bedaquiline. That is not, doesn't have regulatory approval.
So remember, when it gets trial, it only gets trial at a certain age group. And then you go a step lower, and that hasn't happened for years.
I'm Rajneesh from.
Say the same thing actually. Clinically, no, actually, so basically the challenges with the young children always is that clinically you are not permitted to study them at the first time point, and therefore anything that you develop in a clinical phase perspective happens last in young children and in pregnant mothers. So I think there is a huge gap, as you rightly said, and there are opportunities for us to actually really make amends. Of course, the other point that is always looked into is the immune system of the status of the person. And the immune status of young children are very different from an adult, actually, from very many perspectives. So there are clear-cut, gaps that you are there.
Yeah, just to add to that, remember, pediatric is one hand, but the geriatric is also another issue where we understand so little. Other questions?
I'm Rajneesh from DBT. So my questions, I have two questions which are from the general public point of view. First is, it could be a myth among the public. Is pranayam has any impact on TB? That is my first question. Second question is, if we look at the overall treatment of TB, we are considering TB, just like another bacteria with a thick skin. And we are always inventing antibacterials, which are definitely going to be develop resistance. So is there any other alternative therapy has been, tested, tried, like nuclear medicine? Or we have seen in COVID, like there is a sudden rush of cytokines and everything is, it's destroying the lung cells and everything. So why can't we, utilize COVID as a like bacteriophage? Nowadays, bacteriophage is also getting used as a mode of delivering drugs and mode of destroying the bacteria. We have never tested that. I don't know.
So pranayam, I don't know, but I will talk about the fact that there is a lot of emphasis on host-directed therapies for that reason, because you're right that no matter whatever antibiotic we come up with, there is likely to be some resistance, some level of resistance that will develop. So host-directed therapies aim to overcome these because these are going to target the host responses. And two examples today. One was metformin, actually three examples. One was metformin, the other was nutrition, and the third one was sertraline. So there is work being done in that area, and that's very important in TB right now.
Sorry for being disruptive, but we need running short of time. So we'll close the question answer session for this panel. Some of us are around, so please catch hold of them. Thank you.
So thank you to the esteemed panelists from session 1 for joining us. And before we move on to the second part of the panel discussion, I'll take a moment to introduce Dr. Balganesh, the chair. Dr. T.S. Balganesh is a distinguished scientist with a rich history in drug discovery. He led AstraZeneca's R&D, delivering their first molecule for clinical studies in 2007, and was recognized with the Yvonne Ostrom Medal for Drug Discovery. He's headed the CSIR's open source drug discovery program, focusing on neglected diseases. He's a recipient of the honorary doctorate from University of Uppsala in 2011 for his groundbreaking work in tuberculosis and malaria drug discovery. Currently, he's the president for Gangajan Biotechnologies, where for bacteriophage-based therapies for serious bacterial infections. He's also the chair of Special Technical Expert Committee for Indian Tuberculosis Genomic Surveillance Program. So with this, I'll invite the second part of the panel discussants to take us to the transformative power of molecular innovations, artificial intelligence integrated platforms, which will accelerate our progress towards TB elimination. May I request Dr. Anamika Gambhir to welcome the panelists in the second session. Dr. Vinay Kumar Nandikuri. Dr. Shibu Vijayan, please. Dr. Prapti Jaiswal. And with that, I hand over to Dr. Balganesh to continue with the session, please.
Moving on to session B, where we're more focused on some of the things that's happening based on the initiative that Rajesh took in terms of getting more information through sequencing on strains from all over. Also, we've got a distinguished panel of speakers talking on different aspects of TB. So can I first invite Dr. Vinay Nandikuri, immunologist, biotechnologist, and currently the director of the Center for Cellular and Molecular Biology. Is known for his studies on kinase mediated, which he started many years ago, and I actually worked with him at that point. The signaling networks in tuberculosis. He holds the prestigious J.C. Bose Fellowship. So Vinay, all yours.
Thank you. Okay. Thanks, BG. And I would like to talk about some of the things about TB elimination through molecular innovations. I wasn't sure what kind of introduction is required and what can will be given. So there will be some things that may be redundant, but nevertheless, I'll skip that part of it when I get a chance. So we all know this part of it. TB is an important killer, and India has the biggest problem. It's our problem. We need to deal with it. That's an aspect which I want to point out. So in this context, actually, I mean, 2 years ago, I think almost, we started working on what is known about TB in India. The number of, like, we did not even know which clade of TB or what is more prevalent in India compared to the world. We did not know drug resistant patterns, extent of drug resistance, if there are novel mutations that are present in India. So all these things were open questions. And that's when the INTIX part kind of started off, as a multiple effort initiated by Rajesh, and quite a lot of us got into it to do this. So this is also a bigger problem because, if you really think of tuberculosis, we all think of only pulmonary tuberculosis, and we talk only of pulmonary tuberculosis. But look at it, we get in almost every part of your body, and it's almost 15%. Actually, it's not that small. And quite a lot of times when you go to the doctor, it is not even easy to diagnose if you have tuberculosis if extrapulmonary tuberculosis. Some of them are relatively easier, but some of them are quite difficult. For example, if you were to have tuberculosis in your reproductive organs, it's a difficult thing to do. Liver, difficult. Gastro, better than others, but nothing is that easy when it comes to diagnosis of extrapulmonary tuberculosis. Effort done by many people with respect to diagnosis in terms of pulmonary is relatively easy, but in terms of diagnosis of TB, some of them people talked about, I think Urvashi Singh was talking about a little bit about biomarker discovery. The challenge about biomarker discovery and doing, finding out those which are found in TB infection, but not found in other things, is that, other bacterial infections also roughly give the same biomarkers of regulation. That makes it complex. You know that there is an infection, not necessarily an infection because of TB alone. So it's kind of, I think, a bit of empirical treatment is still on, and culturing bacteria coming from other parts of the body is quite difficult. For example, we have a collaboration with the LV Prasad Eye Institute. They get ocular TB, and they were very confident they were Mycobacterium tuberculosis. We ended up sequencing them. Then none of them they were mostly NTMs. They were not non-tubercular mycobacteria. None of them were actually tuberculosis. We sequenced almost 15 samples recently as part of this INTIX actually. So it's very difficult to figure out is because of NTM, the non-tubercular mycobacteria, or is it because of tuberculosis where the infection is where you have sometimes extrapulmonary. So when you start talking about TB as a whole, there are always 4 aspects, no matter any kind of a disease. Diagnosis, which is what I will deal mostly with as time today. Treatment, which Ashwini talked about a bit. And vaccine, none of us did, but really it's an aspect that need to be looked at. And surveillance. So surveillance is closely connected to the diagnosis. So that is a part which I'll deal with today. And vaccine and treatment I'll touch upon, and maybe even skip, depending on, since it's already been talked extensively about it. Vaccine, not as much. Treatment, it's already been talked about. I'm skipping this part of it. So vaccine, right? This is a slide which is a highly, what happens when a person gets infected, how TB gets eliminated or ultimately forms a granuloma, how many kinds of immune responses happen. Now, if you are talking about a vaccine for TB, what is known is BCG, right? Bovis BCG, which is a live attenuated vaccine, works reasonably well in children, but not in the adults. And there are a multitude of efforts that are happening across which are involved all the way from activated TB vaccine, attenuated TB vaccines, recombinant BCG vaccines, like their improved versions, and subunit TB vaccines, DNA vaccines. And one thing that is not really written here, is mRNA-based vaccine. So we are actually started kind of working already in parallel, working on mRNA-based vaccine at CCMB along with the AIC CCMB. This is about actually our idea. I think this is a collaboration with Dheeraj, will be next year. But basic idea is to actually make mRNA vaccine where around 8 antigens of TB are expressed, one shot. And this can be done individually, or at a later point of time, the idea would be to do it along with BCG to see if that is going to work better. This is an aspect that we have kind of initiated. That's why I was saying that there is a molecular aspect even to vaccine development. That's the reason I wanted to talk about vaccine development. Like mRNA completely requires, generation of constructs. Everything is completely molecular. So INTIX TB is a surveillance program essentially. And this is what we aim to achieve, is that can we actually rapidly diagnose TB, MDR TB through genetic acid? This is the bottom line of what we want to ultimately achieve. Can we come up with a point of care diagnostic based on sequencing, which actually can tell the treatment regime for the person who's infected with tuberculosis? We obviously need AI/ML models for AMR predictions, and I think we are going to touch upon it later. Standardization of the rapid and reliable detection of genetic variants. All these things are kind of something that we want to touch upon. This is basically a slide that is missing consortium of so many of us coming together to do this. Whole of report consortia is working on this, and there are so many people who are involved in collection of the samples. Collections come to ICGB. In ICGB, we actually make repository as well as then DNA is made, and the genomic DNA is then sent to CCMB and NIBMG 50/50. And ICMR has its own 5,000, which they are doing. And these samples are then sequenced. Data is sent to NII, National Institute of Immunology, where the analysis happens. We are also planning to do transcriptome data that will come from, again, NII and then CCMB and NIBMG. All of us can be involved in this. It's a huge effort given the fact that many things have to move. And obviously there are logistic nightmares and a few other things here and there. But with all those limitations and all, we wanted to do it faster, but we seem to have reached 10,000 eventually, and which is going to be talked about today. So, but what is that we want to achieve given such kind of a complex project is first thing that we can think of is diagnosis. Diagnosis is the first part. Somebody goes and they have no idea. Doctor would suggest, either a quantiferon or a T-spot, that is what we call as Mantoux test. If they're negative, you're not considered TB. But if you're positive, or if you're a person with a lot of symptoms, then you can have X-ray, smear microscopy, or microbial culture. These are a little bit more classic old style. They work, but not very sensitive. Nowadays, mostly what is given is nucleic acid amplification test, which is mostly expert MTB or RIF or TrueNet and other things are available for people to be diagnosed. In all these cases, you need to have a certain load, and that is true across the board, and extrapulmonary tuberculosis gets missed. So what do we want to eventually achieve is not simply about finding out we have a TB. We should be able to tell what are the resistances, if they are resistant to certain drugs or not. How do we do that? You really have to, if we can actually include next generation sequencing, come up with ways to do this, either using nanopore or at the point of care, then we may be able to actually within a reasonable time frame, maybe say 1 week, we can come up with an idea to say, okay, this is the clade and these are the resistance patterns. So maybe doctors can make a better decision on what would be the better drug combination that can be given to the patient, which essentially eliminates failures. Like after 1 month, they come back, they're resistant to something, those kind of things. So this is the ultimate aim. And this is where our molecular, whatever work that we are doing should come together. This is all that I wanted to talk about. And All the people involved in intakes, including many people are involved in this mega project, and there is a website that looks at it and really gives you an idea, and it tells you one already that has happened. Mostly, if you talk about the world, you say lineage 2. You look at our sequencing data, it's mostly lineage 1 and lineage 3 after that, then lineage 2. And what we need to know over a period of time is, with respect to India, which lineage is prevalent where? Because we know where the samples are coming from. And we also know the drug resistant patterns. So are we going to have some novel mutations which are not identified earlier? If we find them, then we have to go back and do basic research on them to find out how they are contributing to the drug resistance and ultimately make it part of a panel so that we can have a point of care diagnostics. Thank you.
Thank you, Vinay. Let's move on. Can I now invite Dr. Shivu, who's the Chief Medical Officer for Global Health at Cure AI, a leading company that utilizes AI to enhance healthcare access and quality. His expertise in both clinical and public health setting drives impactful advancements. Looking forward to something which is happening, going to happen.
Thank you. Thank you so much for the introduction. Nice introduction. So I was wondering why I'm here. So most of the stuff has been covered today. The only reason I am here is because 1983, I took 18 months of treatment for tuberculosis, and I had the fortunate to write 6-month prescription in the national TB program, which makes me kind of look proud, starting from mass miniature radiography in this country, sputum-based microscopy, and now to molecular diagnosis. And now today we are going to talk about genome sequencing, which is goosebumps moment for me. So couple of slides is purely conflict of interest for me. With what I'm going to present is because I work for a company called Cure.ai, which is essentially an AI company for primarily radiological image interpretation. And we do have a TB product, which is a flagship product, deployed across 50 countries, which helps in triaging, interpreting X-rays and give a probability score. I just mentioned about it. I don't want to eat a lot of time on that. We call it as QXR, which is essentially an X-ray AI interpreter software. Urvashi Madam mentioned about thousands of mass radiography happening at the field with ultra portable tiny X-rays going to the field and then screening the vulnerable population. Also mentioned about she's also leveraging technology and AI for screening X-rays, which is happening in the facilities. So we are one among the pioneers from India, which is validated globally by WHO, reference in WHO documents, just completed the WHO pre call and kind of cleared it. And recently HTA clearance has happened. ICMR validated, deployed in almost 400 sites in India. I'm just skipping it, which is with a lot of promising findings coming out of it. What are the opportunities? Because most of the time, because I've seen TB pretty significantly over the last 33 years, starting with a very rude to a much more smarter bug now. But what is the role of genome sequencing? Because I was in the thick of Mumbai TB control in 2012 when it is totally drug-resistant TB was announced, and then I was charged to be part of a project to engage the private sector, blaming everything to the private sector prescriptions. And now that research shows it's not the private sector alone, we also equally contributed, not from a sector-wide, but also from the type of prescriptions what we make, and also the bug. The unknowns about the bug is probably going to be coming from the work what you just completed. That's going to be immense, because I have witnessed the cry from the Mumbai clinicians and microbiologists saying we need sequencing. And now that, again, proud moment, Madam was saying that TGS is used for treatment decisions. That has been a cry from the Mumbai physicians always. But now that when you're having 10,000 data sets, what are you going to do with that? So that's the task which I'm tasked to do that. So essentially, from a charge, how do you analyze this data? So we have 10,000 data sets, a huge data sets. How are you going to utilize this? this reference panel is going to help us in kind of understanding more. Is it imputation? Can you do imputation, more imputation? But essentially, you are increasing the resolution of the data set you have. Genetic diversity analysis. We did heard about something which is in that direction also going. And we know that there is a predominant Beijing strain, which has been known for drug resistance, which is prominently in the Mumbai region and also in the northeastern region, whereas in South India region, it is Central Asia and African strains are there, which is slightly much more reformed and tamed, I would say, compared to the Beijing stuff, which all these informations were patchy. So now that when we have these reference panels, we could make a meaningful recommendation out of it. Transmission thing was being touched upon from the earlier mentions, but again, we now have a well-reformed stand. We can design transition models. Madam mentioned about the reactivation versus the reinfections thing, because again, molecular studies from Mumbai has clearly demonstrated there is a significant reinfection than the reactivation bit, because the reactivation bit could largely be modified by some of the work you have done, Dr. Sheetal, which you modify your immune response or your host responses, and then accordingly, or even nutrition probably can help in modifying that part. And then, which we have seen significantly, the reactivation bit is coming down. The global evidence also say that reactivation is coming down over the years. Probably the reinfection, given the high burden status, is something which we are not tackled, and that will give you, the studies will give you more insights about it. Epidemiological infection, again, transmission, outbreaks, the patterns across India. Accordingly, we can also discuss about what is the law and vaccine development. Vaccine development has been touched upon here. I'm not going to talk about again. But again, coming to the precision medicine, how do you tweak your treatment, whether it is shorter duration, larger duration, what is the endpoint, what kind of genetic pattern is there to design a treatment which is contextual and customized to a particular bug variant. That also opens up with this pool of information coming out. Drug development, this also is being touched upon by Dr. Balganesh, was mentioned about was challenging as like, are we doing the right thing? Because we have been, as I said, I've seen 1993 onwards, the regimen or the revised national TB program regimen has been tested here, DOT strategy. We have never moved from that to almost be stuck on that for 25 years to actually look at the daily regimen from the thrice weekly to the thrice weekly had a lot of relapse rates, which has been stalking, and now with the daily and DOTs, which has been coming down. And the third part is the vaccine development, which also has been touched upon. I'm just moving on. But as I'm representing from an AI company, I also want to actually mention about how are you going to use the technology and AI on these things. As you know that these are all huge data sets, which is humongous data analysis need to be done. I'm good. I'm actually proud that Pragya is here, Prapti is here, so like she will do a demonstration because she's specifically focusing on AI applications on the sequencing bit. So she will do a demo on that. But again, I'm saying data analysis is something which AI can come in. Pattern recognition, because from the wide set of mutations available, how do you going to make sense from this thing? And AI could be a good tool for it. Predictive modeling, again, this is being used in non-sequencing setting also. So this data, as I mentioned, this data sets. How do you kind of like look at and make this interact with other vulnerability or other indicators, other biomarkers, which is again, we are not so. In tuberculosis, most of the innovations have been tested in silos. If I'm doing one, okay, drugs are done for drug. How do you combine drug plus the bug plus the host responses? Can we, as that is where massive data overlay and insights being derived from that, AI can play a critical role. Integration with other technologies. Can we integrate with the liquid biopsies, targeted genome panels, and monitor genetic changes over the period of time? So I would urge what next, do a detailed analysis. That's something probably will be in the pipeline, and we're happy to be part of such initiatives. Use of this application, most of the time, these repositories will remain as repositories. So just want to kind of like nuts to think about doing epi, drug, treatment decisions, and vaccines kind of studies pivoting on it. Continue to understand, because we need trends, how these patterns are going to change. So because this is a one-time exercise, fabulous, timely done. But again, how are you going to actually continue this thing? Looking at, are we going to establish central surveillance centers so that, and looking at, okay, the change in pattern of genetics, transmission, is there a new bug emerging? All these things are share this data. There is a WHO catalog, which there is an open call for contributing to the global database. So that's something, I mean, Government of India is also making such attempts. I'm hoping that this will probably show up in that also. Research, interdisciplinary, tech-driven, AI-powered. Thank you so much. I'm handing it over to Prapti to come and demonstrate her product. Thank you.
I hope once you've gone through the elimination phase, TB treatment will actually become personalized.
I wish I had a 2 weeks treatment rather than 18 months.
Where we can actually design based on and AI. So thank you very much. Now may I please invite Dr. Jaiswal, the co-founder and CEO of RUG AI. She's merging her experience in molecular biology and microbiology with AI and genomics to combat AMR. Public health issues, all yours.
Good afternoon. Thank you, Dr. Gokhale, for this invite, because it's an esteemed audience and a privilege to be speaking here, especially because before I founded our AI, I was a tuberculosis researcher at THSTI. My supervisor is over here. And the support of ICGB and NII is what built my foundation in the science. And Dr. M.K. Bhan's Foundation Day lecture put the seed of translation in my head, where we want to apply our academic education into real-world application. So presenting to you the next frontier of my career is Arogya AI, where we provide precision medicine for tuberculosis. Arogya AI's core competency is to analyze NGS data for data-driven treatment decision-making and driving all the public health policy and application, as has been alluded to by the previous speakers. What you see here is the genome sequence, the genome sequences of Mycobacterial tuberculosis. And we're searching through with a fine-tooth comb mutations that correlate to resistance within that genome. The gold standard for that correlation of association and causality of resistance through those mutations has been established by the WHO in the form of a catalog, which has been revised recently in 2023. Our cloud deployed NGS analytics solution covers and uses that gold standard for this analysis. And the next frontier for applying artificial intelligence, which is to be oversimplified to be pattern recognition necessarily, where with one genome, 2 genome, and N genomes can together form a stacking of information. And with that, we're able to continuously learn patterns. That continuous learning is inspired by evolution, where the continuous learning can be acquired by machine learning algorithms. And what that machine learning algorithm can then provide is novel findings that speakers have been alluding to that are thus far not known in literature. Yes, it requires validation in silico, in vitro, we'll have knockouts, we'll have complement studies, but the direction is to make it faster, to be able to achieve more information now with cutting edge technology. So a faster path from discovery to the context of why we're speaking over here. The gray pipeline is what you have conventionally for analysis of genomic sequences right now, where from the genomic sequence, there are bioinformatics pipelines that will align and annotate the genome. You look at it, correlate to the catalog that exists, which was previously manually curated, but can be augmented with machine learning and artificial intelligence. Like I said, the data will have to be validated. One of the key questions that the machine learning algorithms can answer is the epistatus and the correlations for that are beyond just their expression, but the correlation of how each gene affects the nature of the mutation and thereby informing resistance. And that is the direction of the company and the ethos of what we're trying to build to inform the different pillars of the public health solutions in tuberculosis. So Arogya AI goes from the FASTQ file, which is the raw data, to a report and a summary for drug resistance in less than 40 minutes. Our pipelines are able to do this analysis. It is enabled by AWS. And the vision is to find and enable physicians and clinicians to make more data-driven treatment decisions, moving away from empirical prescriptions of antibiotics. Our report looks like this, where on the left, you will see the drugs. So this includes first-line drugs, second-line drugs, the newer drugs. All the information about the newer drugs, which is covered in the WHO catalog, is what we have included. And in addition to that, the scientific community will want explainability on why I'm making a claim of resistance, and all of that information is reflected in our reports. In addition to looking at these reports from an individual patient level, we're able to analyze for a population level where we can do batch processing of samples and take inferences with phylogenetic analysis. As the speakers have spoken about the different lineages, where if you see the circled single lineage, which is coming in between lineage, the blue one and the yellow one, is possibly an emergence of a new lineage potentially. And that strain characterization is one of the biggest strengths that machine learning models can impart to something like the InteGS value chain that's being created with 10,000s and tens of thousands of genomes, which will potentially indicate a possibility of new lineages in the Indian populations that were thus far not characterized. That is the scope of the machine learning that can be employed. What Arogya AI has done so far since 2019 is we've done a bunch of peer-reviewed work. The first analysis was against all other existing genomic tools, where we showed superiority to TB Profiler and Microbe. None of those free-to-use softwares were updated by the WHO catalog in 2023. Arogya AI's technology is superior. That gave us confidence to run a clinical validation in a clinical setting at AIIMS Jodhpur with a cohort of about 500 clinical samples with phenotypic culture DST to make a comparison, as well as against the existing technologies. So the first comparison we've shown is against the CBNAT, where the key finding over here is you see the star at the bottom, single star. That is where Arogya AI's superiority in distinguishing between MTBC and NTM is highlighted. There were 66 samples that were NTMs that were erroneously detected by CBNAT as MTBC. So these are the advantages we have over the existing technologies. We were also compared to the most dominant molecular technique in the program at the moment, but the largest limiting factor of that is that a lot of genomic mutations are not covered. That is the advantage whole genome sequencing will have in addition to LPA. And in comparison to the culture DST, we saw high concordance in the usual suspects and some discordance in the usual suspects, meaning pyrazinamide and ethionamide. There was ethambutol, my apologies. We had discordance simply because of the known issues with mechanisms of action and those issues in culture DSTs. So this gave us confidence to do this with a larger sample size of over 1,000 whole genomes to see that there is a reliability in the data that we're producing, and we're ready to deploy and aid the DBT's InteGS in whatever manner, because this is the true translation that we had visioned and I learned about from Dr. M.K. Bhan back when I started my PhD. Here I'm going to demonstrate a video of how it works. The platform is a software, and all of us as biologists have only seen very complicated softwares. So we've built an easy-to-use drag and drop software. Done all the hard work doing the sequencing. Unfortunately, I can't change the sequencing methodology and hardware, but I can make the analysis easier. So if you can press play on the video, please, you'll see that you're simply clicking on our web portal. Drag and drop, we're agnostic to hardware used. So you click Illumina pairwise because that's what we've been used in this example. Drag drop your FASTQ 2, 5, 10, 20, 100 FASTQs in about 25 to 45 minutes. You will have the reports being spit out like that, where you can look at the different microbial diversity. So it'll tell you the NTM, it'll tell you qualitatively and quantitatively which bacteria and species is present within that sample, the quality controls, the drug susceptibility test, and all of this as a combined report with explainability. So we're all here ready to deploy, where there is absolutely no reason towards a TB Mukt Bharat. We have NTM detection. We detect the 3 newer drugs. Batch processing is possible, which is not available anytime until today. It's a one-click report generation. It's a no-brainer to deploy. And while I understand that is an expensive solution, last week at the TB Innovation Summit, my mentor, Dr. Gangdeep Kang, spoke about the economics of scale and how our country demonstrated about a 10x drop in pricing for RT-PCRs in a span of a little over a year. I truly believe we can achieve that with scale for our technologies that is sustainable for innovators as well as for the programs, because our technology and innovation needs to be accessed by those who need it the most. It takes a village to put it all together and build this. There are many people I would like to thank and have gone in to building this out. In addition to the team, we've got grant funding and support from the ecosystem. And because it's World TB Day, there is a free demo for the product. Please scan the QR code, test it out, give us your feedback, and let us work together. I want to end by saying stalwarts like Dr. Balakrishnan, veterans, have spent decades in the space of tuberculosis to move the needle. It's time to pass the baton to us. Our generation is in the interest to move faster and move things on the foundations that you have laid. So thank you again for this opportunity.
Thank you, Jaiswal. Appreciate it, but the baton is yours. All right, thank you very much. We've got some time, so a couple of things we can do is take questions both from panel A and panel B. I know a number of people put their hands up, and I had abruptly said no. So if there are, is that okay?
Yeah, I guess.
If there are questions, so please. Yes, sir.
Hi, Pragya. It was a nice talk. Regarding this app, like you said that we have to upload the FASTQ files. So how is it like translated in the clinical setups? Like doctors and clinicians, they won't have access to these FASTQ files. So how can they like get to know whenever they have a new TB patient come in, like what type of resistance he or she has? So what is your take on this app?
Thank you for your question. My name is Prapti Jaiswal, not Pragya.
Jaiswal.
Okay. The vision is to enable clinicians to make more data-driven treatment decisions. Yes, we are not at point of care. We're at where genomic sequencing and NGS is. At the moment, it is at tertiary, at the state and district TB office care. So we're on the cloud and we plug into the genomic sequencing machine. Anybody who has access to that is going to be able to access the analysis after they've done the hard work of the genomic sequencing. We're only going to be adding to the problem if this becomes an app on somebody's phone, because I am not qualified to prescribe anti-tuberculosis treatment. I am only qualified to do the science and inform the clinician who takes the treatment decision. I hope that answers your question.
But how will you like overcome the problem of the FASTQ file?
What's the
FASTQ file, right? Whenever you have to like get the whole analysis of the strain, you need to have a FASTQ file.
Yes.
How will you take out the problem with having a FASTQ file?
You have to sequence it, right?
I mean, without the sequence, you can't.
I mean, without an input, how do you expect an output?
Yeah.
So, but that part is where we were talking about that, one has to develop POC, point of care diagnosis. We can actually do the sequencing using either nanopore, Illumina, or anything else. We are in conversation to see if we can actually, once we know what are the possible mutations that are there that are responsible for drug resistance. No, some of them are already known, but if more things are known, one can design faster sequencing ways to address this.
Thank you. Yeah, Mohan.
So this is a question for Vinay and Prapti. I'm not sure whether this is quite early, but what is the kind of differences between the EPTB isolates and the pulmonary TB isolates? Is it too early to ask?
Do we have EPTB isolates in our this thing yet? We have. We have not analyzed that part of it yet. So once we know, we will be able to answer it.
I'd like to add to that. So we did a study with Dr. Camilla Rodrigues' group in Hinduja Hospital, and they have sequenced over 150 EPTB isolates, and that entire study is currently in peer review. I cannot say the results just yet, but you will find out. There's a significant, there is some similarity, there is also significant difference. We wanted to prove that our algorithm is able to analyze from both EPTB as well as pulmonary.
As an extension, is it possible from the sequence to know that these basically don't go through the lung towards the other organs, or basically it goes straight? I know it's a difficult question, but
I wouldn't know. Not from.
Dr. Urvashi mentioned something on that. 40% you also see the lung lesion. Which came first, I don't know.
Other questions? Yes, please. Training data sets, okay, for analysis. Now, how many number of cases you have taken for the training purposes, for application of AI or ML?
When we started, we had less than 100. Today, with the global data sets, there's about 27,000 training data that we have used to build our models. However, I think with the INTGS, there is a case to be made in the context of the Indian tuberculosis genomes.
In that 27,000 of training data set, how is it translated to how much of a sensitivity or a specificity?
I'm very glad you asked that question, sir. So, sensitivity and specificity are actually the wrong measure in this context. Concordance and discordance are a better measure simply because we use sensitivity and specificity when we're trying to identify a diagnostic tool. Maybe yes, no. However, here, because of the incidence of drug resistance for each specific drug is different. So we had a slide where the clofazimine was 1 in 400 in that set. If I were to do a table for specificity and sensitivity, the values would be extremely skewed. But we are able to do that same work with concordance and discordance. So by and large, we're seeing above 85% concordance in comparison to phenotypic DST with its caveats. However, a genotypic correlation also is done to cover for both because the gold standard varies. If the gold standard is going to be phenotypic DST, then against that a concordance will be measured. There are also technical parameters of the machine learning model, which is accuracy, precision, and reproducibility that are built in, and the rigor is then measured against the clinical samples.
Yes, please.
Hello, my question is directed to Dr. Vinay Nandikuri related to diagnosis of the TB. So like there is asymptomatic patient or latent TB patient. So what kind of sampling you do for this kind of patient sample collection, and what kind of efficient test you have to detect those TBs?
As of now, mostly it is Montus test or IGRA that is used for the latent.
Sir, mic is off.
I said either IGRA or Montus test are used for the latent tuberculosis patients. I mean, are people. And in fact, that is used even for the household contacts when suppose you know somebody has TB and if the other people have or not, they actually use this at a first level to quantiferon, which I was saying, or the Montus test, which is again PPD is essentially proteins. So culture filtrate proteins of tuberculosis at a certain dose are injected under your skin and you look at response. That is the way they actually define. But otherwise you don't have TB there, right? To culture or do anything with.
Yeah. Okay.
Just wanted to complement that because the report study is also doing a long-term follow-up of subclinical tuberculosis. So very recently WHO has redefined subclinical with the microbiologically confirmed or microbiologically unconfirmed subclinical tuberculosis. Essentially, X-ray is a tool to flag, both the microbiological and non-microbiologically confirmed. But they're also looking at biomarkers to kind of jointly how this prediction goes up. Like X-ray is one part of it. Then you are adding metadata from the patient, whether it is, other symptoms. So let's symbol like a C-reactive protein, ESR, and other host, and also complex things like a host response being measured with other parameters also being looked at, and joint indicators like a radiological marker plus biomarker, how it is performing. So it will take some time to get into the right answer. Then how is it the subclinical is going to be diagnosed? It will take a significant time. So we are part of a research study in UCT as part of the report, but again, it will take some time to throw some light.
Yeah, 1 second. Dr. Sonali, you want to add
So
on?
if I may add, actually subclinical TB is one which is microbiologically confirmed TB. So we what usually do is chest X-ray, of course, but that's not microbial confirmation. So bronchoalveolar lavage, so or induced sputum. So they may not be having cough, but when you induce, they produce some sputum, and that is microbiologically positive. That is subclinical TB.
We need to close. It's a short question. Go ahead.
Yeah. So the question is for all. So is it like our long-term goal is to take the sputum sample and from the sputum sample harvest MTB, take material, put it in the sequencer, get the FASTQ file, and then on the same day or maybe in 10
Same day
days.
is a stretch.
So is it like in 2 weeks time, like
About
after
a week's time,
expansion?
one should be able to do it end to end.
Okay. Yeah, that will save lots of time. Yeah. Thank you.
Okay. So I'd like to thank all the panel speakers of both panel B and panel A. Thank you very much.
Thank you.
Great learning experience.
Thank you, sir. So we could probably have a group photograph for the panel B also, sir.
Sorry?
Just a group photograph for the second session. Sir.
Yeah.
So on behalf of the Department of Biotechnology, I would like to extend a very all the panelists to having agreed to come here on this occasion. And we'll soon be commencing with the second part of our session. And Honorable Minister is going to be here shortly. We request you to please be seated and
Something else. Something from that. They said that is it prime time? Yes. What makes
And they've all spoken as a photographer after that just all over and have a picture with this. I'm not sure. Yeah, but
In the past decade, Bharat has emerged as a powerhouse of economic growth under the able leadership of Honorable Prime Minister Shri Narendra Modi, showcasing its resilience and potential on the global stage. Now, as we stand at the threshold of a new era, there's a transformative opportunity on the horizon. Biomanufacturing. Biomanufacturing holds the key to unlocking the regenerative and sustainable potential of biological systems, paving the way for the national priority of accelerated green growth.
भारत को ग्लोबल ग्रीन एनर्जी मार्केट में एक लीड प्लेयर के रूप में स्थापित करने में भी अहम भूमिका निभाएगा।
To steer Bharat towards this vision, the Union Cabinet approved an integrated bio policy, biotechnology for economy, environment, and employment. Bio-E3 policy was formulated after a series of dedicated sectorial meetings with multiple stakeholders across the country. The BioE3 policy will bring together scientific and technological advancements to enable circular bioeconomy, facilitate scale-up of bio-based products, streamline policy regulations, expand their market opportunities. The policy envisions to establish a robust framework for sustainable growth by intensifying entrepreneurship, public-private co-creation model, and setting up of the Molankur Bioenablers Hubs. The policy harnesses the potential of biotech engineering and digitalization to spark innovation, nurture skilled workforce, and boost employment. With an estimated $2 to $4 trillion of direct annual global impact, the BioE3 policy represents a monumental shift towards efficient, tailored production process and sustainable solutions for Viksit Bharat. Join us in shaping a greener, more prosperous future. Together, let's harness the power of biomanufacturing to build a sustainable tomorrow for Bharat.
Okay.
Chairman, Secretary Secretary.
In India, the production of ethanol from creates large amounts of spent wash, a dark brown wastewater containing hazardous melanoidin. The spent wash is a significant environmental challenge. Melanoidin in the water inhibits photosynthesis, making it unsuitable for irrigation and difficult to treat in wastewater systems. But there is a sustainable solution. Algae biomass can be processed through hydrothermal liquefaction to produce biocrude oil and biochar. Biocrude oil can be refined into biofuel, while the biochar can play a crucial role in cleaning spent wash. By passing spent wash through a biochar filter, hazardous melanoidin is removed, making the water safe for irrigation, thus reducing its environmental impact. This innovative approach turns a waste problem into a valuable resource, supporting sustainable agriculture and a cleaner environment. The leftover biochar after filtration can be added to compost to produce new biofertilizers that fix carbon in soil and enrich soil fertility to improve crop productivity. Cleaner water, better crops, and a greener planet, all through the power of algae and biochar. In the past decade, Bharat has emerged as a powerhouse of economic growth under the able leadership of Honorable Prime Minister Shri Narendra Modi, showcasing its resilience and potential on the global stage. Now, as we stand at the threshold of a new era, there's a transformative opportunity on the horizon. Biomanufacturing. Biomanufacturing holds the key to unlocking the regenerative and sustainable potential of biological systems, paving the way for the national priority of accelerated green growth.
भारत को ग्लोबल ग्रीन एनर्जी मार्केट में एक लीड प्लेयर के रूप में स्थापित करने में भी अहम भूमिका निभाएगा।
To steer Bharat towards this vision, the Union Cabinet approved an integrated Bio-E3 policy, biotechnology for economy, environment, and employment. Bio-E3 policy was formulated after a series of dedicated sectorial meetings with multiple stakeholders across the country. The Bio-E3 policy will bring together scientific and technological advancements to enable circular bioeconomy, facilitate scale-up of bio-based products, streamline policies and regulations to expand their market opportunities. The policy envisions to establish a robust framework for sustainable growth.
Good morning, everyone. Our chief guest for today's program, Honorable Minister, has arrived at the venue and will be with us shortly. I request all members in the audience to kindly take their seats, switch their phones off, and quickly settle down, please. So good afternoon, everyone. Our chief guest for today's program, Honorable Minister, is here with us. May I request the Honorable Minister to join us on the dais, please. I humbly request Dr. Rajesh. Yes, sir. This side. I humbly request Dr. Rajesh S. Kokle, Secretary DBT, Dr. N. Kalaisalvi, Secretary DSI, DG CSIR, Dr. Rajiv Behel, Secretary DHR and DG ICMR, Dr. M. Srinivas, Director AIMS, to please join on the dais. So on behalf of the Department of Biotechnology, I extend a very warm welcome to our chief guest, Honorable Minister of State, Independent Charge, Science and Technology, Minister of State, Prime Minister's Office, Personal, Public Grievances and Pensions, Department of Atomic Energy and Department of Space, Dr. Jitendra Singh Ji, in this program marking the World Tuberculosis Day. May I request Dr. Anamika Gambhir, Advisor, Department of Biotechnology, to present a gesture of welcome to Honorable Minister Dr. Jitendra Singh Ji. I request Dr. Anamika Gambhir to please present a welcome token to dignitaries on the dais. Dr. Rajesh S. Gokhale, Secretary DBT, DG BRIC and Chairman BIRAC. To Dr. N. Kalaisalvi, Secretary DSIR and DG CSIR. To Dr. Rajiv Behel, Secretary DHR and DG ICMR. To Dr. M. Srinivas, Director AIMS, All India Institute of Medical Sciences. Thank you, ma'am. I now request Dr. Anamika Gambhir to kindly deliver the welcome address.
Yeah, this is it. Of State Independent Charge, Ministry of Science and Technology and Ministry of Earth Sciences, Minister of State for PMO, Ministry of Personnel, Public Grievances and Pensions, Department of Atomic Energy and Department of Space, Government of India, Shri Jitendra Singh Ji, Secretary DBT, DG BRIC and Chairman BIRAC, Dr. Rajesh S. Gokhale, Secretary DSIR and DG CSIR, Dr. N. Kalai Selvi, Secretary DHR and DG ICMR, Dr. Rajiv Behel, Director AIMS, Dr. M. Srinivas, esteemed delegates, distinguished guests, and my dear colleagues, BIRAC and our autonomous institutions. On behalf of the Department of Biotechnology, it is my proud privilege to welcome you all to this program to mark the World TB Day today. I would like to extend a very special thanks and gratitude to our Honorable Minister, Sir, Dr. Jitendra Singh Ji, whose unwavering support, decisive leadership, and consistent encouragement has always been the guiding light for all of us as we work towards utilizing science and technology to find meaningful solutions for the largely needs, especially the health. So the World Tuberculosis Day, as you all know, is a day dedicated to raising awareness and advancing our efforts to combat one of the world's most persistent and deadly diseases, tuberculosis or TB. So after having a hard-hitting session since morning, hard-hitting science since morning, let me just take you through this simple stuff and take you through the history of TB to mark the significance of this day. So tuberculosis, as we all know, has plagued humanity for centuries, and it continues to Is this some problem? And it continues to devastate millions globally, inflicting severe health, social, and economic consequences. So as we delve into the history of TB, find that it was known to ancient civilizations with mentions by Aristotle in 322 BC, Hippocrates in 460 BC. It also finds a mention in the book of the famous Arab scientist Ibn Sina. Of course, then it was known as phthisis or phthisis, which actually means a progressive disease, since the term TB was coined much later. In fact, so ancient is this disease that its evidence is found in mummies from the prehistoric times as well. So by the 1800s, TB had become one of the most feared diseases, with 1 in 4 people dying from it. In the late 1890s, it still claimed 1 in 7 lives, and for much of history, it was a leading cause of death worldwide. Historically, this disease was known by many names. Consumption, because of the severe weight loss it inflicted on the patient. White death, because of the extreme pallor seen in those infected. Pott's disease, named after the surgeon who first described tuberculosis of the spine. And phthisis, which means a progressive wasting disease. So some of the early advances to combat the disease happened in the year 1834 with Johann Lucas of Wurzburg, when he coined the term tuberculosis to describe the affliction of the tubercles. Then on 24th March 1882, a major breakthrough streptomycin. As we fast forward the time to decades later, after an era of astonishing scientific and technological advances, proactive government policies and support, we have made significant strides
in understanding and treating TB, something I think is witnessed by most of us in this room. And although we have made significant progress in our understanding, there is still a considerable challenge ahead of us. I think all present here can appreciate the fact that in the recent years, India has made significant strides in combating TB. The TB incident rate has decreased by 17.7% from 237 per 100,000 in 2015 to 195 per 100,000 in 2023. TB-related deaths have also declined thanks to the fresh impetus by the Pradhan Mantri TB Mukt Bharat Abhiyan. Although we had pledged a significant reduction in TB by 2025 ahead of the SDG target of 2030, the COVID pandemic derailed our efforts and the progress, causing increased vulnerability to the disease. There were major disruptions, diagnosis and treatment, and of course, a significant resource and funding was diverted to the management of COVID. But nevertheless, we are over with the tough times of pandemic and have again intensified efforts in the direction to regain our momentum. Now coming to the DBT initiatives in this direction, well, at the Department of Biotechnology, we've always given importance to research in driving innovations and finding sustainable solutions to combat TB, and have been funding TB research since the inception of our department. We have prioritized research in several key areas, which include research on the biology of Mycobacterium tuberculosis, development of newer diagnostic tools and techniques. Significant investment has also gone into understanding the mechanism of drug resistance, exploring potential drug vaccine candidates and novel adjuvants for the TB vaccine. I would also like to make a mention of the notable initiative in this regard, which is Report India, established under the Indo-US Vaccine Action Program. Since 2013, this collaboration between DBT and the US National Institute of Health has set up 7 clinicals across the country. The TB consortium that it has created involves long-term cohorts of TB patients and their household contacts. This has provided critical data for advancing our understanding of TB and its transmission. So another very significant initiative is the Dare to Eradicate TB program, launched on March 24, 2022. Under this umbrella program, data-driven research is employed to address TB eradication. The Indian Tuberculosis Genomic Surveillance Consortium is also a part of the umbrella Dare to Eradicate TB program, under which we are sequencing the genomes of over 32,000 TB isolates. And as part of this program, we are celebrating a significant milestone today of having sequenced 10,000 genomes. CSIR and ICMR institutes are also part of this effort. This effort will provide invaluable insights not only into the genetic diversity of Mycobacterium, but also on the evolution of drug resistance in TB. Additionally, we are also focusing on extrapulmonary tuberculosis under Dare to Eradicate TB. Research is underway to develop tissue-specific treatment regimen for EPTB, taking into account the unique characteristics of sites such as spine, nervous system, and gastrointestinal tract. So I'll conclude here while reaffirming our commitment to the fight against tuberculosis, together through science, collaboration, and innovation, and with the constant support of our Honorable Minister, Dr. Jitendra Singh Ji, whose leadership has been instrumental in advancing our TB research initiatives, we will continue to push forward in our quest to end this ancient scourge. Thank you once again, and a very warm welcome to each one of you. Thank you.
So thank you, ma'am, for quickly taking us through the history of tuberculosis from the early civilization times to where we stand today on this occasion. For the keynote address, we have amongst us an eminent and highly respected medical professional who's not only known for his compassion and empathy, but also for advancement of medical education and research, as well as building healthcare infrastructure. Please join me in welcoming Dr. M. Srinivas, the Director of All India Institute of Medical Sciences, for the keynote talk.
Thank you for the kind words. Respected Dr. Jitendra Singh Ji, Honorable Minister of State, Independent Charge, Minister of Science and Technology, Minister of Earth Sciences, Minister of States and the PMO, Minister of Personnel, Public Grievances and Pensions, Atomic Energy and the Space, Dr. Rajesh Gokhale Ji, Secretary DBT, Regional BRIC and the Chairman of the BIRAC, Dr. Rajiv Banerjee, Secretary DHR, DGICMR, respected Madam Kalai Ji, Secretary DSIR, DGCS.
The experts from all parts of the country. Truly, sir, I must thank you because this disease needs a collaboration. And the collaboration I can see here between not only the ministries, institutions, people, the experts and everything, because this challenge cannot be met by one agency or one lab or something. So truly, your leadership and your guidance and the leaders who have conceived this and then brought all the way today is something which is commendable. I must congratulate the leaders and your leadership, sir. Thank you very much. Well, I've been asked to speak on the keynote address. Experts have already been deliberating since morning. I had an opportunity to discuss with, sit there and then listen to the people there. The theme today is, yes, we can end TB, commit, invest, and deliver. That's what the key words have been. This is the commitment which we need to have. We are showing it here. We have all the people, you know, the best of the people in this country. All the experts in the field of tuberculosis are here. So the investment has happened. You know, you can see that the Honorable Minister is going to talk about 10,000, the sequencing and everything. The delivery is an issue. There are issues with that, and then that's going to be delivered and that's going to be discussed and delivered. So it's very aptly there that on this day of World Tuberculosis Day that we are assembled here to see what is that which we can do together. The fundamental thing is the togetherness of all the people who are in this country and the policymakers of the. Some of this has been already spoken in the morning, and I don't want to repeat that. What is the burden which we have? This is the global burden, the statistics which have come from the World Health Organization, that look into the burden of the disease, such a huge burden of the disease, and then significant of that comes from the Indian burden. You know, 28% of that is something which we are contributing to the world. And when you look into that areas where the Southeast Asia and the part of the Africa, that's where the disease burden is going to be, is very high. We need to focus on these areas where we can do a change. Then WHO and TB strategy, the milestones have been put. And I think we need to do a lot of introspection on that. You know, 2025, are we going to deliver what we are supposed to deliver? I think it is up to the leadership and the people, if necessary, the course corrections. What is that we can do? Truly, those milestones which are there, which has been spoken since morning, it's something which we need to keep in mind. The youngsters need to keep in mind that, you know, we have a bigger mission to go with this. You know, this is an easy job for us. And these things should be kept in your mind that, you know, as a country, as a team, all of us, are we going to, you know, do this and then be part and parcel of achieving this, not only at the world level, what the WHO set in and what we are set in for 2025. Well, since morning, we've been talking about the various newer diagnostics and all. There are people here that even today that there is a lot of discussion is happening. The very fact that a lot of discussion is happening means that we need to really those things, what is the best suitable for our context in our country. And since morning, we've been looking at the molecular diagnosis, the neurodiagnosis. This is again, as a keynote address, I just want to bring it to your kind attention that, you know, there are many things which we need to do. Are we going into the district level, the district hospital? Are we going up to the primary level hospitals? Are we going to address the drug resistance in a way where the technology can bring the things to that level? You know, that's something which we need to look into that as a team. Country has a huge burden. We may not have the experts in everywhere. You know, look into the thing. You know, are we having that kind of radiologist in all nooks and corners of the country? Is it possible to take up that kind of huge load of the thing? And we just heard from Madam that, you know, how the AI and the machine learning tools are going to help us in triaging and then diagnosing them, early diagnosis in a large scale. And that's definitely something which we need to look into that, not only at the radiological diagnosis, but also of the the microbiological diagnosis. That is something which we need to look into. And then prevention plays a very important role for us because there are people, they may not be aware of the tuberculosis, but they are part and parcel of propagating the disease in our communities. Again, that we need to focus on that and look into that, especially the people with HIV. Then what is that which we can do? Can we bring a newer guidelines, management of tuberculosis prevention treatment in India? These are the certain things which I'm going to, flashing here so that we can look into those things and then learn them. Vaccine, well, some discussion has already happened in the morning. A long way to go. And this, happy to say that, many institutes are working on that. Definitely, we should have something. And the AIIMS has been part and parcel of that. I just want to share with you that, AIIMS Delhi was the site of ICMR TB vaccine regulatory trial. The trial ended in a few months ago, and soon it's going to be published. Maybe the DG sir is going to allude on that, the various, what are the outcomes of that. The impact of different intervention to reduce TB in incidence and mortality. Well, the focus has been on the nutrition because morning also we heard from many experts that, it is not only increasing the incidence, but also the chances of propagating. And in fact, we go back to our olden days, when we were students, the TB sanatorium, what did they give? They give the nutrition, they give good air, they give good sunlight. And then that made a huge difference in that, That definitely has a huge role to play, the nutrition. And the curves, the outcome curves with respect to various initiatives which we can put, and especially the component, the last area of the component is the nutrition. Are we going to change? Yes, definitely the nutrition is going to have a huge change in the outcomes of the tuberculosis and this is just to summarize the whole thing and what is that which we can have, what are the best things in the rankings, I put it in that way, that what is that first, second, third, fourth, and fifth, which highest of the intervention with respect to the value addition of that, preventive therapy, that's definitely number one. TB candidate vaccine, definitely it's there. The BCG vaccination, again, it is there. It is useful, especially in the babies, small children, especially for decreasing the morbidity. You look into the central nervous system and other things where there are a lot of complications. Definitely it has a role to play. Then nutritional therapy, which we discussed in the morning and then being discussed again in the later the discussions in this meeting. Targeted screening therapy, definitely that has a role. And then the various things which have been put up. This is what we have compiled from the various studies which have come up through. And the prevalence of pulmonary tuberculosis in young adult patients with type 1 diabetes. Madam spoke, Professor Gurshi, in the morning about that and what is that we have contributed from All India's Medical Sciences to the existing literature and where do we go. And these are the challenges. If I have to sum up in the keynote address that these are the challenges which we have, these are the challenges which we need to look into that so that we will work together in a collaborative fashion to make that, what we are saying, the TB Mukt Bharat. So this is what is the various ministries and the government organizations are working on that. The 100 days intensified TB Mukt Abhiyan has been successful. We have heard the very successful stories and that's where we have defined our goals and objectives and the burden of the disease in the country when you look into that, where the various districts which have, and the focus has been on those 347 districts which are high disease burden, that's where the focus has to happen. And the various campaign activities which are happening as not only today, but it's an ongoing activities which the ministry has taken. The many ministries, all the ministries are working on that. Various screening, low node testing, the notification, differentiated TB care, nutritional prevention of the TT treatment, various protocols which are being discussed, all that. So these are the various methods where we are going to look into that. The expected outcome of our 100 days intensified TB Mukt Bharat Abhiyan is going to end up in how we are going to reduce our the rates and then contribute to the overall targets which we need to achieve by 2025. And then to achieve the TB Mukt Bharat, then we need an increased access to the high quality care and early diagnosis of tuberculosis, especially with respect to the MDR TB, and improve the diagnostic care, the treatment completion. This is very important because
the completion doesn't happen, then there are issues. It is not only with respect to the complaints, but also with respect to the resistance and also the recurrence of the disease. Active case finding, again, is a high burden setting at risk population. We need to look into them and active case finding and treatment of latent TB. This is especially very important with people with HIV. And of course, nutrition has been alluded to in the morning, and vaccine definitely when available at the population level. That is going to be a big game changer.
I just wanted to sum up the things to say that, All India Institute of Medical Sciences is definitely committed. And I'm very happy that today the collaboration is not happening just between the institutions, but between the various institutions of the various ministries here and the various sciences, whether it is the molecular, basic sciences, or the clinical sciences. And the theme which says the commit, invest, and deliver, we are part and parcel of each one of them. And it definitely shows our commitment. For the past 2 or 3 decades, we have been an active in this, not only the diagnosis, but also the treatment protocols, the guidelines for the country. And then I'm happy to share with you that we are one of the WHO centers for training, teaching, policy, advocacy on tuberculosis. This is something which I wanted to share with you from the All India Institute of Medical Sciences. And I once again congratulate the leadership and the visionary leader and the various people who have been here. And together we can make the TB Mukt Bharat, and then that togetherness I can see here. And I'm very optimistic about the fact that, together, yes, definitely we have made a beginning. And let us work together so that we become part and parcel of that successful story of TB Mukt Bharat. Thank you very much.
Thank you, sir, for sharing your valuable perspectives. And we are certain that with commitment and collaboration, we can achieve the ambitious goals we have before us. We request the representative team from Indian Tuberculosis Genomic Surveillance Consortium Program to share their journey from the inception to where we stand today. And for that, I hand over to Dr. Devasis Mohanty, Program Coordinator for the program and Director, BRIC NII. Over to you, sir.
Thank you, Neha. Honorable Minister, Dr. Jitendra Singh Ji, Dr. Gokhale, Dr. Rajiv Bell, Dr. Kalai Selvi, Dr. Srinivas, all the dignitaries and the faculty, students and all. It is indeed an honor to present the progress of INTGS, Indian Tuberculosis Genome Surveillance Consortium. And this is, as was told before, it is a power of use the power of genomics and AI to tackle the tuberculosis and to be part of the ambitious goal of the Government of India for TB Mukt Bharat. So the main purpose is how we can do diagnosis of drug-resistant tuberculosis faster by using the genomics and AI. And of course, as was told, any progress, any project of this nature, which is so ambitious, will not be possible to do by one institute. It requires participation from many institutes, and we learned it from our COVID experience. And like in SACOG, this is a project which has like 3 of the DVT institutes, NII, ICMR, ICGB, and many of the ICMR Center, CSIR, and Ministry of Health. And so here, and our INTGS portal was already announced released by Honorable Minister several months back, and there was a commitment there as and when all this data will be constantly released through the INTGS portal. So all the data on these 10,000 genomes and the drug resistance profile is available on the INTGS portal. But more than what is important is to share how we reached there. And as was told, it requires the data has to come from the clinical centers. The sample has to come. There was the report consortium of DBT, which DBT has been funding through the Indo-US vaccine program. And ICMR has its program to monitor TB and sequence all over the country. So ICMR NIRT has contributing to it. So Dr. Sonali and Dr. Sip Kumar would give an overview of those work. And after that, the unique to this project is we have built a strain repository at ICGB. Like any mutation, if anybody wants, will be able to get from the strain repository and work on it. Dr. Dheeraj will talk about it. And then it will be Dr. Anis will talk about the sequencing work which is going on at CCMB, NIBMG, and also at NII. And at the end, I will just come to give a little bit overview of the AI/ML applications. So I would request all my colleagues to be brief so that we can stick to the time. So Dr. Sonali, please.
Thank you very much, sir. A very good afternoon to all, Honorable Minister, dignitaries on and off the dais, and dear friends. So before we go into the details of the bug, which you're waiting for eagerly, I'll take you briefly over the details of the patients who were infected with this bug and contributed to the sequencing data. So starting with the Report India consortium that was mentioned about, 5 sites in Report India, that is PGI Chandigarh, Hinduja from Mumbai, JIPMA Puducherry, BMMC Hyderabad, and BJGMC Pune, almost the whole of geographical, area in India. So it was a pan distribution of sites. And the list here you see under the major NTP partners is not exhaustive. It is some of the places which contributed to the major bulk of the patients that we got the samples from. With the support, of course, of the state and the district tuberculosis offices in each of these states and the districts, we were able to achieve what we have achieved today. The intermediate reference laboratories were the crux program. So we started with our data from collection of excess of the samples that were actually collected for the routine tests in these labs. And we also collected the AP data and the phenotypic data from the Nikshay portal that is maintained by the ministry. So these samples are stored at the IRLs, then shipped to the Report India CRS clinical research sites. And then from there, some initial processing and culture were sent to ICGEB for the DNA extraction, and then the further institutions had the WGS done. So again, briefly, the patients who contributed to this data. The characteristics of the patients, we see that most of our patients were in the 18 to 45 years age group, and there were children and also elderly. And my apologies for an error here. It is the male which contributed to 68% and a female 31%. And most of them belonged to the BPL families, and majority of them were, almost all of them were EPTB, but we have some of those samples from the EPTB patients. So this actually represents the patients in the country. So the samples that we got, the patients we collected the samples from are representative of all TB patients in the country. It's a very representative group that we have got. The symptoms among these patients, many were asymptomatic, which they did not report. They have cough, but they do not recognize it as a cause of, because of TB. So that is why this high proportion, as you can see, as asymptomatic. But there were other symptoms as well. But here is the importance of subclinical TB, as we have been discussing since morning. The other comorbidities, again, we have had lengthy discussions in the previous panel discussion. Burden of HIV among our TB patients is thankfully very, very low. So very few were HIV positive. Diabetes, again, we had discussions. It's quite high a burden among our TB patients. Underweight, again, 50%, similar to what was reported by Dr. Sheetal. And this is the similar, everywhere across India and in our consortium, the TB patients, 50% of them were underweight. Obese were few. Smokers, again, about 20%, and alcohol use was found among around 18%. So there were other tests also performed on those samples that we collected for the WGS. So PNAT, TrueNAT smear, and so these actually help us to get even the DST and culture help us to get the phenotypic data for our patients. So I'll just end with the treatment outcomes, because everything finally ends up with what happened to those patients who were infected with these bugs. Unfortunately, we could not, this data did not help about 7% of these patients who died. Probably this data that we are generating will help us get some diagnostics and later on help appropriate
and personalized treatment for patients, which will reduce the death among TB patients. But in our sample, we had about 80, about 60 and here 20, about 80% cured and treatment completed, but we did have deaths and other loss to follow-up and adverse treatment outcomes as well. Here I'll let Shiva take over.
Good afternoon, and Honorable Minister Ji and respected dignitaries. So I'll be presenting the sample collection strategy, which was followed by ICMR and NIRT for this study. And this study was funded by, this part of the study funded by ICMR totally for sample collection as well as for genomic sequencing and phenotypic testing. So the difference was all the samples which are collected as part of this this part was done phenotypic testing as well as genotypic testing, and it's still going on. So the strategy was that we continuously collected close to 5,200 samples, that is over a period of time. And this was done directly from the district TB centers, and these were cultured in IRL. So all there is a mechanism already built in wherein all the positive samples are shifted to the IRLs, which are intermediate reference present in each state. And these samples were cultured, and this culture positive samples were then transported to ICMR and NIRT for sequencing. And at ICMR and NIRT, we did both phenotypic, that is using culture-based DST, drug sensitivity testing, and also the genotypic DST. So the strategy was also included collection of samples from around 276 districts, 28 states, which were distributed in 6 groups. So to ease the collection and follow-up of collection. And these state groups were also linked with NRLs so that the follow-up of samples can be done smoothly. So a total of, this is the distribution of sample in the study across India, and we have collected, you can see the red dots represents and where the samples were collected, and the number is denoted in the first, second column, and the collected samples is denoted in the third column. And the sample collection was little more than what is required because some of the samples were directly transported as sputum or processed sample. That's why the numbers are more. So progress, we have done around 5,200 isolate or the process sputum collection, and 4,800 midget or the liquid collective. Out of that, around 2,000 midget DST has been done for 10 drugs, and these 10 drugs include first-line drugs, second-line drugs, as well as newer and repurposed drugs, so that we can build a catalog of mutations for even the newer drugs for a better sensitivity and specificity of these drugs in terms of concordance also. So, and we have sequenced around 4,000 sequencing has been done, and further it is going on. Thank you.
Good afternoon, Honorable Minister Ji and all the dignitaries around. The whole program was being discussed and evolved. One of the important aspects of it was to ensure that there is a uniformity when we are targeting 30 or more than 30,000 samples to be handled, in terms of DNA extraction, in terms of sequencing, in terms of how they are handled at the site where these clinical samples come. And one thing that really helped at that time was having this tuberculosis aerosol challenge facility, which is the DBT supported BSL-3 lab at ICGB, which we have been operating for almost now more than 16, 17 years, and has been serving to different TB researchers from all over the country. Having that facility at ICGB allowed us to plan in a way that we can operate in a hub and spoke model where samples from different clinical sites will arrive, will be processed in a uniform manner, and will extract DNA, which will be sent for sequencing to the sites like CCMB and IBMG and to some extent at NII. But also it will allow us to kind of store those clinical isolates in near native format, in the sense that you are not really doing repeated cultures, So you are storing in the way they are coming in, and they just develop the stocks and keep it in a deep freeze for whenever you need. Anyone has any question related to a particular mutation, one can go back and isolate, restart the cultures and look at that. And that's how this whole protocol was designed, that we receive the samples, we assign it a specific INTGS ID, and once those INTGS, once those IDs are assigned, then half of it is just taken forward for DNA extraction. A part of it is cultured into a liquid broth, which is then stocked into into these microbeads, which is for long-term storage inside -80 deep freezer. The genomic DNA that gets extracted goes to CCMB and NIBMG and NII, where the sequencing takes place. And we recently completed the sample that's received at ICGB BSL-3 lab to almost 10,000 samples. The total numbers are more because we have NIRT samples also. Those samples, once arrive, the biorepository numbers will go further high. And it doesn't take much to really understand that how important it is to have this biorepository. Also, this is one of the biggest single site storage of these many clinical isolates, that too well characterized with such high depth DNA sequencing data which is available for them. So it's going to be humongous and very precious resource that we are going to develop as part of this INTGS program, and which I think is going to be something which will certainly help in taking the field in a momentous way forward. So that's status, and right now we are operating at a at a very high speed. We can do 500 to 1,000 genomic DNA extraction in a week. The biorepository is going a little slow because the major emphasis has been on getting the extraction and genomic sequencing part to be done. But repository also we are catching up, and we are about just crossed 5,000, and those are well stored and available. And what I've done is just put some glimpses of people working inside BSL-3 lab and how those biorepositories looks like at different stages of development. As you culture them in liquid culture, you take out the DNA, extract DNA, and then put them back into -80 for long-term storage. So these are things I just put forward for everyone's understanding. And I think once these DNA are sent forward to the sequencing side, that's where the job of NII comes, when they start getting the DNA sequence data. And now I will request Anish to take it forward in terms of diagnostics.
Good afternoon to the Honorable Minister Ji and all the other dignitaries on the dais and the audience. So I'll briefly talk about general statistics about the sequencings that we have done. So yeah, before getting into that, I just want to also emphasize, so we have sequenced like 10,000 new cases, collected. We started the collection in February, little over 12 months. We collected 10,000 samples. We cultured them. These are very slow growing organism, and sequenced them and analyzed, and it is accessible on the INTGS portal for people to see. So I really want to emphasize on the hard work a lot of people have put into this whole program. Now, the sequencing, the main sequencing centers are CCMB, which has sequenced around 3,800 samples, and NIBMG, around 4,600 samples. NII, NIRT, and Hinduja also have contributed significantly to the whole effort. And briefly, this is the bioinformatic pipeline that we use. We map using Bautype2, variant calling using FreeBayes, and mostly I want to also highlight here the AMR prediction was done using the traditional tools like TB Profiler and the WHO catalog, and also an in-house developed AML model. So Dr. Mohanty will about it in detail a little bit later. So the overall stats of the sequencing is matching with what we know about the distribution of different clades in India. Like we have the lineage 1, the highest, then followed by lineage 3, and we have a significant fraction of lineage 2 as well. So that's kind of matches with what we already know. And when we look into the drug resistance pattern among these strains, we see that around 24% of the strains we got have some kind of resistance, with 7% showing single drug resistance and around 17% showing resistance against multiple drugs. I mean, more than one drug. So, of course, that seems to be higher, but there could be some sampling bias here because we need to get the consent forms and bunch of things are there. So some amount of sampling bias also will be there. It will be corrected as we finish the study. We know the outcome of the patients as well. And briefly talking about, this is the lineage-wide distribution of different resistance. So the Beijing strain or the lineage 2 is more associated with drug resistance. That's exactly what I'm seeing here too. The yellow one, the lineage 2 has more prevalence of resistance here. And this is the distribution of multiple, I'll just skip this part. And finally, the most important achievement of this program will be, once it is finished, is going to be this portal, the IntelliGS portal, which is freely accessible to all. Right now, also, it is fully accessible, where we can look for the drug resistance data. For example, if you go to the browse and look for a particular antibiotic, you can get all the data about that antibiotic resistance. And here you can strain is resistant to both ethanamide and isoniazid. If you click on that particular strain's IntelliGS number, you can get all the data about that, including the DST, the phenotypic DST and the genotypic DST. And we'll add more data, including the disease outcome. All this data will also be added to this portal. Now, Dr. Mohanty will talk about the next part, which will give a sneak peek about why this study is important.
So once you get the genome sequence, of course, we want to do a drug resistance prediction just based on the genome data. If you see the type of tools which are available, one is WHO mutation catalog, which is a manually curated, very rigorous data based on 50,000 genomes, TB genomes all over the globe. Like some tools like TB Profiler will be a rule-based method. And like, let us say there are many machine learning methods. In this project, we have developed a machine learning method called TB-AMR PRED that can also deal directly with FASTA sequence. It can deal with the reads, FASTQ files, and do a drug resistance assignment. Machine learning methods have the advantages they can take into account real time. You do not have to wait for WHO to come to this one. But however, most important is all these tools have to be validated. So to validate the tool, you require, of course, a phenotypic DST. With that, you can compare. So as part of the IntelliGS project, we had data about 918 samples for which we had genomes and we had the phenotypic DST result. And in another project, one of the IntelliGS PI, Dr. Camilla, had contributed to the cryptic that had about 1,500 Indian sequences, and that also had the drug resistance profile based on MIC. So we took all these 2,400 data together. Now we tested all the tools, machine learning, WHO catalog, and rule-based methods. And as you see here, and this is like certain antibiotics, the results are encouraging, like let's say isoniazid, rifampicin, but there is still significant deviations in terms of like false negative prediction. A sample which is told as actually experimentally found to be resistant, but these tools are not able to detect. And there are some new antibiotics. Of course, you have more deviations. But what is important point is all these tools which has been used, they are trained on the global data, where Indian data is underrepresented there, and that is the purpose of a project like IntelliGS. Like this highlights the need of India-specific data has to go. That is where we can do better. So this is what is showing this. The regions where we failed, where are the false negative, what it has is it has many mutations based on global data. They have been ranked as not associated with resistance because they are occurring in the susceptible samples in also large numbers. So if you put them into resistance, what will happen? Your false positive cases will go on. But nonetheless, it is important to understand why that mutation is causing this one. So this is where a correlated analysis of mutations can help. Like this mutation is present in combination with some other mutations is making one sample resistance, whereas in combination with some other mutation, it is susceptible. So this what we intend to do, and all this, what it shows is any such new mutations or these things, these are not sequencing artifacts. This tells you about the what are the like sequencing depth, average depth, and all these things. So what we hope to do in future, based on this analysis, we will be discovering this newer mutation go to the profile, which will be, that is what is the main purpose of the project. And with this, and what next point, what it has already been discussed is, even if we manage to do all this, because we are doing a culture-based method for genome sequencing, the time frame of all this pipeline is going to be 3 to 4 weeks. You will not be able to reduce the timeline of a culture-based method. But what it has given us is, it has linked for which antibiotic, which genes you are seeing mutations. Using this, if you develop targeted NGS, and this will help us, this data will help to design the gene panel which can be used for targeted NGS. Dr. Vinay talked about it earlier, and targeted NGS is also part of WHO recommendations, and Dr. Urvashi also mentioned about it. So what we hope to do will be, this project will contribute information based on which targeted NGS panels can be made, and this could be lead to, in that case, we might be able to do in the clinic with a time frame of 1 week the diagnosis of a drug-resistant tuberculosis. With this, and these are the outcomes of this project, we have a pan-Indian collection of genomes and drug resistance profile, and India-specific reference genome and mutations, and we can update the mutation catalog routinely as more data comes, and developing targeted NES, and also, as in the first two talks, Dr. Sonali and Dr. Siva said, we have a lot of clinical data which can be correlated with this. And I have to thank all the people, of course, DBT, all the CSIR, ICMR, and especially I have to thank Dr. Anamika, Dr. Jyoti, Dr. Vasnya, and Dr. Neha. They have really cooperative, and this is, without them, this project would not have been possible. And other than the people who presented, every center, a large number of other people are involved. I have to thank you. I'll stop here. Thank you.
Thank you, Team NTGS. We are confident that the project will maintain its momentum and deliver impactful real-world applications. We now have a brief showcase that will take us on a visual journey through the NTGS program.
एक बहुत बड़ी चुनौती का मुकाबला कर रहा है। ये चुनौती है टीबी की, जिसे क्षय रोग भी कहा जाता है। भारत ने संकल्प किया है 2025 तक टीबी मुक्त भारत बनाने का।
India is estimated to have the highest global burden of tuberculosis, a disease caused by Mycobacterium tuberculosis. Growing drug resistance in Mycobacterium tuberculosis and transmission is a significant barrier in controlling tuberculosis. In line with Government of India's mission, TB Mukt Bharat, the Department of Biotechnology, Ministry of Science and Technology, and Ministry of Health and Family Welfare have jointly set up the Indian Tuberculosis Genome Sequencing Consortium, the INTEX. INTEX is a network of 9 collaborating institutions across India. Samples collected from each center are processed at the clinical research sites of the Report India network. The consortium has so far sequenced 10,000 mycobacterium genomes and is cataloging the existing and emerging resistance mutations through genomics and artificial intelligence. Whole genome sequencing and targeted sequencing detect resistance mutations within hours to days as compared to the traditional culture-based drug susceptibility testing that takes weeks to identify resistant TB strains. Genomics-based approach through INTEGS, therefore, has the potential to transform the course of TB epidemic via early and accurate diagnosis. Understanding the distribution of clinical strains in the population, as well as that of drug resistance patterns in association with treatment outcomes, may help in better diagnosis and foster a move towards more community-based management of drug-resistant TB.
Thank you. So moving on, just a round of applause for the video, please. Moving on, may I invite Dr. N. Kalaiselvi, Secretary DSIR and DG CSIR for her address.
Namaskar, Vanakkam and pleasant afternoon to each and every one of you. Most respected and Honorable Minister of Science and Technology, esteemed leaders on the dais, and very dignified experts of the dais, researchers, well-wishers of this particular program. I'm extremely happy to be here because 3 years back on the same day, when Honorable Prime Minister announced that we should have a country which should be totally free from tuberculosis, that is TB Mukt Bharat. Now, after the period of 3 years, today we are able to come up with we are able to showcase some kind of a success with which we are standing here. Though we have committed for about 30,000 plus kind of sample collection, now we are done with 1/3 of it. And therefore, let us just make it as a point of celebrating this success and taking this as a part of the success. I think we will have more confidence and more courage to take it further in the way it is expected to be taken in the coming times. And I'm just given to understand that once upon a time, we thought that even 10 million people, the death rate it is only 1.5 or 15%. If it is not treated, this tuberculosis is going to take the surveys or the surviving of the people to the extent of even 50%. Therefore, just to put a full stop to that particular threat, I think this activity is very important. I wish to give a great salute to this particular of people because you have come up as one consortia and one team under this day to eradicate TB. I think I appreciate this is a time that all of you should be deeply appreciated. So 10,000 sampling have been already completed. And I'm given to understand that this kind of a sequence and this reference, it is going to be India specific. Like the way it was explained, we need not get carried away by the kind of the reference of other countries. We have to come up, whether it is even shoes, even Indian shoes, we are now coming up with a kind of a India model and India standards. And therefore for disease, we should have our and this program is really giving a kind of a base by means of which India-based standards are going to be made available. And I'm so happy. Once upon a time, there was a blame to scientific ministries that we are working in silos. Honorable Minister always makes sure, and he is always making us to think that what are the best methods by means of which all of us can collaborate? Sir, now 2 ministries are collaborating in this case, and under each ministry, number of organizations, we are joining hands together. Samples are collected at 9 different sites. It is coming to Delhi, then it is going back to Hyderabad, again it is coming back to Delhi, and finally the data is made available on the portal. I think this is one great consortia, one great team. So I really appreciate each and every one of you. Special congratulations to the Secretary DBT, Dr. Rajesh. And I should not stop here without appreciating Dr. Srinivas, the Director AIMS, as well as my good friend Raji Behel ji. Without all of you and your support and your team's continuous efforts, we may not be able to come up with this kind of a success story by means of which we can make our minister also to come up with a kind of a proud statement that Ministry of Science and Technology is not only talking, we are able to do that also. Thank you so very much. All the very best. Thank you.
Thank you so much, ma'am, for those encouraging and inspiring words. We now request Dr. Rajiv Behel, Secretary DHR and DG ICMR for his kind address.
Honorable Minister of Science and Technology, Shri Jitendra Singh ji, Rajesh Gokhale ji, Kalaiselvi ji, Srinivas ji, all the esteemed dignitaries, I want to my delight, congratulations to the entire consortium, because this is a great example of collaboration and that we are seeing here between different departments, institutions, ministries. And this is the way to move forward as one Team India. TB is a very important disease. We are making impressive progress as we go towards elimination. However, it is a very difficult foe. Why is it difficult? Because it has learned to hide macrophages. You can suppress it, our immune system conquers it, and yet it can get reactivated. You can cure the disease and you can still get a recurrence. So between latent TB, asymptomatic TB, symptomatic TB, recurrent TB, it is a maze through which we have to fight it. And the only way I think we can move forward is through research and innovation. That the power of the research and innovation, I want to highlight 3 or 4 things. We all knew that we needed active case finding, but the program could not afford active case finding because people would not even come to big hospitals or where there were X-ray machines, etc. But now, with the work of our innovators and departments of science, ministries, scientific departments in different ministries, including ICMR, now we have handheld X-rays, which are at a cost that is about 1/2 of the cost that is globally available. And we think very soon we'll be having them at a cost which is 1/4 of the cost. Now we don't need to read an X-ray to call it normal or abnormal. We have multiple Indian AI-based tools that have been validated and are able to now being used in the program. And the 100-day program, they were used to show that to just identify the abnormal from abnormal X-ray, so that you can immediately then move on and do a nucleic acid amplification test or a NAT. And there also, from something which used to cost 2,000 rupees to do one test, I'm really proud to say that today we have a test that costs between 100 rupees and 500 rupees, two tests. 100 rupee test can detect, it's an open PCR test. Every COVID PCR machine can be used to detect TB. So we have reached this level of not only this gives us the ability and gave the program the ability to do active case finding. For latent TB, we have also, in addition to IGRA and the old Montu test or tuberculin skin test, we have the which is much more specific in terms of it can work even when you have the BCG vaccine. Nutrition, we have understood through research the importance of nutrition in that how much reduction in mortality it can give to people who have TB. And you have seen yourself that it's already part of our program. The Honorable Prime Minister has now, and the Minister of Health has released about doubled the amount of the support. About 1,000 rupees a month is given to all TB patients to improve their nutrition. Not only that, but if we showed that if we improve the nutrition of the family members, it reduces the risk of TB by half. So you can cut the risk of TB by half. It's like more effective than any vaccine that is on the anvil. So all these innovations, and was very happy. Last week, we had the India TB Innovation Summit, and there were more than 200 innovations. And these innovations and the innovators, the way they are moving ahead it was so heartening to see that how much we can do, whether it is targeted NGS or non-sputum based diagnosis or AI based tools which innovatively detect TB. there were so many of those innovations in detection, in treatment, in rehabilitation of TB that we saw. So I see this opportunity of having the 10,000 MTB gene data becoming available as another tool through which we will be eventually able to support the program. And I hope very, very quickly we will be able to bring this to the usefulness of the TB elimination program. Thank you very much and congratulations again.
Thank you, sir. Now invite Dr. Rajesh S. Kokle, Secretary DBT, DG BRIC and Chairman BIRAC for his address. And as most of us would know, he is himself an astute researcher in the DB space. Over to you, sir.
Thank you very much, Neha. Honorable Minister of Science and Technology, Dr. Jitendra Singh ji, my secretary colleagues, Dr. N. Kalai Selvi ji, Dr. Rajiv Bahel ji, Director AIMS, Dr. Srinivasan ji, Dr. Anamika Gambhir, Dr. Devashish Mohanty, coordinator of this program, esteemed delegates, distinguished guests, ladies and gentlemen, and my colleagues from DBT, BIRAC institutions and BRIC institutions, actually. I think it's a wonderful occasion to come and announce 1,000 completion of 10,000 genomes primarily. I believe this is from two different reasons. One, as a country, we came together during COVID to really put our resources, ideas, and people together to come up and do activities. And we always said that we can do it in wartime. Can we do it in the peacetime as well, actually? And this is always a challenge. When the peace starts to happen, everybody starts to disperse and they all start to go into their own areas, actually. And during COVID in INSACOG, we realized that we sequenced about 3.4 lakh genome sequences of COVID. It was again coordinated by Department of Biotechnology, but again, ICMR, CSR labs were ahead in every part of their work and activities and contributed to continuous surveillance, actually. And this was very useful because this allowed us to map out what are the strains prevailing around, how does their transmission progresses, what are the kind of mutations that are being arisen. And therefore, that has now allowed us to understand that the COVID now actually has substantially diminished to an extent that it has got, the rate of mutations on its own, it's a kind of have reduced the morbidity and mortality actually. That's not as happening with tuberculosis. It's very interesting to look at, actually. The tuberculosis is such an old disease, have survived for years. However, there is no kind of reduction in its genome virulence with time. And that's the question that also we need to understand biologically. And it causes severe morbidity and mortality in our country. I mean, the numbers are very staggering, as we have just heard from all of you, actually. However, what we need to appreciate is that since the government announced in 2018, actually, TB Mukt Bharat, despite the COVID time, and we should say despite the COVID time, many of us, lungs have been affected during COVID infections, and therefore the susceptibility of TB infections goes high. We've kind of been able to bring it down to 17.7% of, which is twice that of the international standards of what incidence is. So India has done tremendously well in a short period of time, despite all sorts of challenges and problems, actually. And I think this program, therefore, is again another milestone towards contributing, as I say, towards developing a molecular test for rapid diagnosis of TB and drug resistance, as Dr. Mohanty and all the other colleagues talked about it, actually. I think it's very important because what we should understand is that it's constantly we are talked about that TB is a curable disease, actually. However, the moment you get drug resistance, there are challenges that start to emerge, and therefore your ability to improve yourself by taking all sorts of drug combinations and variety of activities does become a challenge to look at. I think I must first of all, sir, remind you that you announced this program 3 years back in the Vigyan Bhavan Hall on 24th March of 2020, actually, on a bigger Vigyan Bhavan Hall, actually. And, and I think the INTGS program, the Indian Tuberculosis Genomic Surveillance, the evidence-based regimen for treating extrapulmonary tuberculosis, and the host-directed therapies as adjunct anti-TB therapy for shortening therapy, actually. All these 3 programs we have started and are ongoing on very good track, actually. And today we are completing one of it, actually, which is very significant milestone. I think I must thank the people who initiated in DBT, Dr. Jyoti and Dr. Vashne, who initiated the and Dr. Anamika and Dr. Neha is continuing these activities, actually, for taking up so much of an interest to be able to do these activities. As you would realize, the clinical sample collection, data collection is not an easy activity as well. And clinical partners who have worked all across the country, Dr. Sonali is here and various other our report cohort members are there, actually, because you have to reach out to people sometimes to be able to collect the samples. So you have to collect these in microbiological, and then the microbiology and antibiotic resistance tests are done on it. And then the isolation of DNA, for example, there are challenges where the BSL-3 shipments were denied by the airlines to be transported across our country. And, we had to reach out to our airlines ministry to be able to tell them that this is packed in boxes that does not allow the BSL-3 kind of material to be transported. I think this is what we are trying to do. We're trying to build new ways of how research will be done in this country, the best practices that are practiced all across the world with the best quality data and the highest quality of analysis that is very, very critical and important for Viksit Bharat perspective. I think if you want to grow as a country, it is very important to develop standards. It is very important to develop confidence and very important for the world to develop confidence in your data that this is what everybody should look at. And I think that's where this 10,000 genome sequence data will be available for everybody to look at, because this is the best in quality. We often actually talk about some works that are done here and there, and they kind of always have a challenge of looking at the quality of the data, quality of analysis, quality of sampling, quality of the phenotypic analysis and data available. And this is what our country requires. And I'm very, very proud of all the consortium members and everybody who has participated in this activity. I'm really thankful for you for actually taking up this challenge. So there are several knowledge gaps in tuberculosis even today, actually, scientifically. And one of the biggest one is the morbidity, sir. As patients recover, the challenge really is in terms of the cachexia that they develop, in terms of their lung functions, in terms of their ability to, get the water retention capacity, the protein to fat ratio content in their body. All of these get altered as you go into the therapy and go through this disease patterns. And this leads to various manifestation of other chronic diseases, which are also quite problematic. And in fact, we are addressing this problem along with the Ministry of Ayush, because we believe that some of the rasayanas that are available actually can in fact improve your quality of organ development and activities. And in fact, the program has already been, will be launched actually. I think part of the activity has already been started, where we have initiated the activity of putting the clinical cohorts together and also testing it out. It's going to be across 9 centers in the country. And CCRS is going to be, sir, partnering in this activity actually along with us, DBT, to do this work. Finally, sir, I must thank you for your time and your always enthusiasm and energy for department's activities. And I think that makes so much more difference to our motivation and allows us to actually bring forth the best from our own perspective. And I think your guidance is always very, very useful, sir. And I know you have a special interest in disease biology, being a clinician by itself, so that makes my life very easier. So it becomes very easy for me to explain or need not even explain to sir what you really need to do actually. And I think the more suggestions I get from you, it's very, very important. I also thank Rajiv ji, Kalai Selvi ji and Srinivas ji for coming over here. I think it's important. The strength of our critical people is really the key thing that we need to do. And finally, in the end, I will tell you one thing, as Dr. Bal Ganesh always says, actually, I think TB is an Indian problem and nobody is going to solve it. Nobody's going to solve it for us. We have to do it. We have to do it our way. And we have to make sure that we provide leadership to this country so that we make sure and ensure the future of our country is much brighter and much bigger. Thank you very much.
Thank you, sir. We're sure we're marching towards TB Mukt Bharat with this. We now have a flyer that encapsulates the program's vision, goals, and the collaborative spirit that drives its success. May I request the dignitaries on the dais to unveil the flyer? I also request Dr. Anamika Gambhir, Dr. Jyoti Logani to join them on the stage, Dr. Devasish Mohanty, coordinator for the program, and the the investigators to kindly join the dignitaries for the unveiling.
Collaboration.
I take more. Thank you, sir. I now humbly request our chief guest for today, Honorable Minister, Science and Technology, Dr. Jitendra Singh Ji, to formally announce the completion of whole genome sequencing of 10,000 isolates of Mycobacterium tuberculosis. So this is for the... So we have a curtain. So I think we can just announce.
Okay. Already Zoom we have been announced. Secretary, Department of Biotechnology, Dr. Rajesh Gokhale, Director General, CSIR, Dr. Kalai Selvi, Dr. Behel, Dr. Srinivas, Dr. Anamika, Dr. Mohanty, the entire team associated with this, members of the fraternity, ladies and gentlemen. I don't have to repeat what has already been said. The burden of tuberculosis has been reiterated again and again. As was being mentioned also by Dr. Srinivas in the beginning, that we have about 1.8 crore tuberculosis patients as by the 2023 data, and the mortality is as much as more than 10 lakh or 1 million. But what is important for us to understand is that the WHO target is to reduce 80% of the prevalence and 90% of the mortality by the year 2030. So it's important to understand the seriousness of the responsibility that we have taken upon ourselves of having a TB Mukt 5 years clear than even the 90% target of the WHO. And for that, of course, this collaboration is imperative. So I'm glad to know that we have in place the DBT, the CSIR, AIMS Delhi, ICMR, and as Dr. Kalai Selvi was saying, 2 ministries in general. And that speaks of whole of science and whole of government. But is this collaboration good enough? That is the question. Because in spite of that, in the last 8 years, if you take from 2015 to 2013, the decline has been only about 17 to 18%. Which means if you have to move on effectively, you have to have not only whole of government and whole of science, but a whole of nation. Unless you involve the common people, raise their consciousness, involve or evoke their participation, you're not going to succeed. And therefore, I sometimes feel the ambitious target may be not that easy. Of course, we would wish it to meet it and also live up to Prime Minister Modi's vision and also the task that he has assigned us. Now, some of us have seen this transition of the tuberculosis profile happening over the last several decades. As researchers, as practitioners of medicine, when we started off in medicine, it was both an enigma as well as a stigma. And if ever anybody had tuberculosis, it was taken as a death alarm unless otherwise. And virtually it became a part of the social parlays, even cultural parlays, even literature parlays. If you see some of the old fiction novels or fiction movies, if you had a triangular love story, to eliminate one in the end, they would conveniently have tuberculosis afflicting one of the characters. What I'm trying to say that it became so much a part of our life that it was like that. And of course, by the time we stepped in, some of the antibiotics had come in, but the sanitoria concept, as Dr. Siri was saying, was still prevalent. In fact, I had the privilege of having done my internship in one of the most reputed tuberculosis sanitoria, that is TB Sanatorium Tambaram, which is very close to the Chennai airport. Kalai Selvi must be familiar with that. No, not that she's been there, but she's seen it from outside because she belongs to that place. Because Asia was considered to be, the biggest reservoir of tuberculosis, and the Britishers very thoughtfully created sanatoria all over the country. And mostly they found locations in the hill areas, open areas. And of course, since Calcutta and Madras were their initial destinations, so we had one of the biggest ones there. We had huge greenery and trees, and even the attendants would come to meet their patient or their relatives who were there. They would leave the food stuff at a distance. So much was the scare. The relatives also would not meet them from a close distance for the fear of getting the droplet infection or whatever. So it was like that. So sometimes they would create a kind of an enclosure. Any attendant coming there would leave that stuff, homemade food, etc., and quit from there. And then, of course, it was reported in 1940s, but it became available only in the early 1950s, the first generation tuberculosis, streptomycin, isoniazid, and then, of course, subsequently. And the stigma was that those days you had very few number of post-graduation seats in the country. So I'm one step senior to Srinivas, so I think by his time things became easier. And we didn't have this common institution that. So after your graduation, it was a practice. You just hovered all over the country to look for a post-graduation. So I was going different places. I went to Rohtak, that Dayanand went to Gwalior, of course, Madras, I was based out, all sorts of places, PGI. And then my guide told me, see, it's very difficult these days to get an MD in medicine. It was indeed, actually. Some of us were very lucky to have ultimately made it. So he said, instead of wasting time, you go in for a diploma. And in fact, diploma was a very common practice those days, postgraduate diploma. You had in all the departments. You had in gynecology, you had in pediatrics, you had in different areas of medicine. There was something called DTCT, Diploma Tuberculosis and Chest Disease. So he said, you go in there for that and then keep trying for the MD. So I apparently agreed. Then went back to the hostel and came back next morning. I didn't know how to face him. So I said, sir, I just want to make a small confession. can I go for some other diploma? He said, why? This is a very practicing branch. I said, no, I come from a rural background and I would be known as a TB wala doctor and nobody will come to me for consultation. So he said, okay, boy, if you are not comfortable. And so then I was toying with other areas, but meanwhile I got into this where I have. But having said that, what I'm trying to say is there's been a long journey. And of course, it was a big ceremony examining a patient of tuberculosis, whether it's fibrous, because as was being mentioned by Dr. Bahel also, I think Mohanty also, nowadays you don't even depend on the clinical examination. But it was a great skill percussing this chest and finding out whether it was fibrosis or whether it's a cavity. If it was top, it was tuberculosis, or down, it was pneumonia. So all those, tidbits were to be learned in your clinical training. Of course, all that became obsolete. That has been taken over by technology. Even Monto's test is no longer very relevant. But the tuberculosis stays where it is. So the point I am trying to make is, I think as researchers, we have to ask ourselves, only 2 weeks back, we had this World Obesity Day. And it started only about 2 decades back, and incidentally, I've been associated with it most of the time. So I was one of the keynote speakers on the International Obesity Federation. So I asked them, I said, every year on this day, you pledge to reduce the obesity prevalence. And every year you come back with a reason prevalence. So either something wrong with us or something wrong with obesity. For example, today, this year, the latest achievement is that India has emerged rank 3 in childhood obesity. On the one hand, you have the city of Delhi full of different kinds of dietitians prescribing variety of formula, from intermittent eating to intermittent fasting, for not eating, for fasting, for whatever I know about. And at the same time, the prevalence is also going up. So, and that on a serious note, I think that is something that we have to ask ourselves. Are we somewhere lacking in our strategy? If not, are we unable to engage the stakeholders and elicit the kind of response that is expected of them? So now, of course, since we are into an era of genes, earlier on we were doing it otherwise. We were still holding awareness camps for tuberculosis. Since my practice was mostly confined to diabetes and endocrinology, so we were holding camps over there also. And there also the prevalence went on each year. And that's why I used to say, the Mark Twain once wrote in the context of economics, he said, economics too serious a subject to be left to economists alone. If you leave to economists, the policy planning, he'll make a mess out of it. So similarly, any disease for that matter, which is in an epidemic form, like tuberculosis, or even for that matter, diabetes is now, is too serious to be left either to a clinician or a medical professional or a biotechnologist. It has to engage the common person, the common man. Unless we are able to do so, we will not, as I said, the level of our approach changes. There were times we were doing mass X-rays, and the chest specialists would take fancy, they would have small reels rolled down in the background of a view box, which was handy. There came a time when you started getting handy view boxes, they were placed on their table. From that, we graduated to genes. But what is the outcome happening? On the other hand, as Rajesh was saying, it was considered to be disease of the Oriental countries, a tropical disease. And that is why till many years at AIMS, we used to get interns from United Kingdom. So you could spot them from a distance, flock of white boys, girls, young, moving together. So you could make out these are interns. Why don't you get them now? Because after HIV, they also had an upsurge of tuberculosis. Total democratization of tuberculosis. So it became universalization. A disease which the Britishers called the disease of the unhygienic people. uncivilized. They would rather talk contemptuously about such diseases, leprosy, malaria. In fact, I remember once in 80s, towards the end of 80s, when we started having an upsurge of the metabolic disorders like type 2 diabetes mellitus, hypertension, coronary vascular disease. So somebody wrote that small humor piece in one of the journals, the Journal of Association of Physicians of India. He said, now it has stepped into an era of civilized. So we can tell them, look here, we are also dying. In the same manner you die. We also die of heart attack. We don't die of tuberculosis. We don't die. But I think that distinction is also over now. Now they are also heading. Now you are having prevalence of tuberculosis may not be to the same extent, but there in West, which was not there at all. Which means the problem is too big to be actually dealt in the manner possibly we have been dealing with it. And in fact, now we have a biphasic disease spectrum. We have the communicable disease like tuberculosis, and we also have the non-communicable. At least in the 50s and 60s, we didn't have coronary heart disease. We didn't have type 2 diabetes mellitus. The burden on the physician was very little. Then after 80s or 90s, we thought now we had got rid of tuberculosis. We were talking only about these metabolic disorders. And suddenly COVID exploded that bit. In fact, one day at the AIMS itself, before you joined, I was telling some of the boys there, I said, sometimes I feel guilty. The teachers of my generation spent more of time emphasizing metabolic disorders. We thought infections were over. Unlike our times, where our teachers spent more time on infections. And now it is just the West. So in fact, I was addressing the residents. I said, they have done a commendable job to reorient themselves to the changing spectrum, which has happened all of a sudden after the pandemic. So now we are beset with a larger reservoir of disease share, and upon that, of course, the lifespan has also increased. And I think the more emphasis now has to be on the preventive health care. Earlier on, we thought we had tuberculosis, then the challenge would be to prevent the complications, which of course still is there. But how best? Because on the one hand, we are talking of Viksit Bharat. We are talking of 2047. We are talking of, we are calling upon every citizen to contribute his energy to the making of New India. So can we allow this energy to be squandered this way? And then, I don't know whether some of you would have noticed, there is a as I said, we have two sets of diseases, both community. There's also a reciprocal relationship between the two. A loosely controlled patient of diabetes or loosely closed glycemic level is more prone to tuberculosis. And a patient with tuberculosis is more prone to diabetes. So God is so kind. He doesn't deprive you of one. It's not that you are now preferring dal, so you will not get paneer. Or if you ask for one, he says you'd have both. If you've got diabetes, you have an easier chance of getting tuberculosis. And that reciprocity also is a challenge. So while treating one of these two conditions, we have to guard against the other. And of course, the only answer is a very strict surveillance, a very strict control. And how that happens, and then of course in this country, we have problem of compliance also. You are giving free medicine for tuberculosis. Have you gone and checked whether he has taken it or not? So how would we do it? So unless the nation's conscious, then there is also a problem of socio-economic reasons. Of course, now under this present government, the personal intervention of Prime Minister, we are providing free of cost medicine. But some of the patients are not going even to collect free of cost medicine. You know why? Because he is registered, for example, at AIMS. He comes from a distance. He's a daily wager, charging 1,000 rupees. Coming and going back, he will spend more than that, or maybe lose 1 day's wage and spend 4. So he prefers to purchase it from nearby. And while purchasing it from pocket, he compromises. He makes, I'm telling you the practical things because I've been dealing with almost 200 patients every day till I got into this. So what he does, he prepares a calendar. 2 days in a week he will miss. Particularly for, of course, tuberculosis, so 6 months, but in cases like diabetes or other things where you take a lifelong, or 5 days in a month, because he has to pay the monthly installment of the flat also. He has to pay the child's fee also. So it's a huge, huge socio-cultural, economic, the multifactorial problem. And therefore, I'm sure next time when we have a program like this, we would also try to involve some of the representatives of the civil society who could actually become our messengers. We are already sensitized in this room. So we are sensitizing the sensitized. So, because you are telling, you are, distributing knowledge to those who know, not to those who don't know, and not even those who don't know that they don't know. So next time, I'm sure we will.
Vector. And meanwhile, we have to, as I always say, you can't leave everything to government. We have to engage the private parties, which has been doing in other programs of biotechnology also. We need to involve the pharma houses, at least those who are in the business of manufacturing antitubercular drugs, so that they could help us in overcoming the social economic factor. So next time we could have a larger consortium of such stakeholders also when we observe the tuberculosis day next year. And I hope we come back with lesser prevalence. Thank you.
Thank you, sir. And your continued guidance is a beacon, and we are confident it will inspire a wealth of future success stories, and will come back next time with a larger consortium and the stakeholders alike. I now invite questions from the press, if any. So we had invited them. So
Good question.
Mike.
I think the whole idea really is to try and identify mutations that leads to drug resistance. क्योंकि सबसे बड़ा चैलेंज जो टीबी में इस वक्त है वो ड्रग रेजिस्टेंस का है. वैसे तो आप अगर आपको टीबी की बीमारी होती है तो आप ड्रग्स लेते हो तो आप ठीक हो जाते हो, लेकिन अगर आपको ड्रग रेजिस्टेंस बैक्टीरिया है तो आपको ड्रग्स को मॉडिफाई करना पड़ेगा रेजिमेन. तो ये किस तरह से जल्दी से ढूंढ सकते हैं, इस तरफ हमारी ये कोशिश है. और इसीलिए क्या करेंगे कि म्यूटेशंस आइडेंटिफाई करके उसके लिए स्क्रीनिंग पैनल बनाएंगे जो इंडिया स्पेसिफिक स्क्रीनिंग पैनल होगा, so that those mutations can be mapped out very quickly and the drugs, appropriate drugs can be given.
Okay, so
Mostly जो resistant वगैरह है ना म्यूटेशन, उसमें इसका ज्यादा फायदा रहेगा, to put it in the simple language.
So we can, so now, since Honorable Minister has to leave for the Parliament back, we'll just speed up with the session. I request Secretary DBT, Dr. Rajesh S. Gokhale, to present a token of our gratitude to Honorable Minister. So
they're calling me there. They said I had to be there too.
Thank you for joining us. So it's.
Thank you, sir. Calling me the Parliament. Please.
So our heartfelt thanks to Honorable Minister for honoring us with his esteemed presence despite his busy schedule, and for his steadfast support for all the department activities. We express our immense gratitude to Secretary DBT, DG BRIC, and Chairman BIRAC, Dr. Rajesh S. Gokhale, for his constant support, invaluable guidance and inspiration, and most importantly, for leading by example. It is not often that we see the key architects of 3 different departments together. The presence of Secretary DSIR and DG CSIR is a testament to the importance of this gathering. We express our deep